A very big hello to my wonderful champions. Just give me a second. Hello, dear children. Am I audible? Am I visible? I'll just wait for some time so that everyone else can also join this session. So we all know that tomorrow is our biology preparatory examination. It's Habiba. Very good evening, dear. A very good evening. So good to see all the familiar people out there. So dear children, all of your friends who also have biology examination tomorrow, share it with them so that we all study together. How are you all feeling? Are you tensed? Well, do not worry about anything, okay? This exam is going to be super good. And over here, let me tell you, I will make sure that I complete the important chapters. The most important chapters would be covered up over here, starting with molecular basis of inheritance, because I feel that it's one of the toughest for you and also one of the most important chapters out there. Yes? So shall we begin? If yes, just give me a thumbs up. If everything is good. Can I go ahead? Okay, so I'll go ahead. So over here, ma'am, duration of this class, I'll try to keep as short as possible. But over here, I am there to at least cover the important chapters, at least. By important chapters, I mean molecular basis of inheritance, principles of inheritance and variation, and also human health and diseases, and sexual reproduction in flowering plants, and also this chapter that is evolution. So evolution is something that we missed the previous time, and I do not want you guys to miss it right now. And also one very important information for every one of you, that is, my dear children, if at all you have seen our channel, okay, if you see our channel and if you go to the video section, over here, if you move a bit down, there is a very important video that I did for biology. I'll show it to you right now so that you do not have any confusion. Yeah, that is the video. Biology Preparatory 70 on 70 Part 2. So this Part 2 contains all the very important questions of the chapter starting from microbes in human welfare. Okay, microbes in human welfare, biotechnology and its application, biotechnology principles and its processes, and what do you have? Organisms and population, biodiversity, ecosystem, everything. All these chapters are covered in this video. Please make sure you watch this video. It contains all the 1 marker as well as 2 to 5 marker questions which have come from these chapters that I have mentioned over here. So this is a previous video. Go watch it. It is definitely going to help you out. Now over here, as I said, I'll start off with molecular basis of inheritance. Okay, Sneha, good evening, dear. Chalo. So let's go ahead with the first question that we have, and sorry, the first concept that we have, and that is organism and their genome size. Remember, you might get match the following question on the basis of this. So this should not be missed. Bacteriophage 5 X174 has the least amount of genomic size. It is just 5386 nucleotides, not beyond that. Which organism has the highest genomic size? Us, my dear children. We have 6.6 into 10 to the power 9 base pairs. Okay, that is the diploid content, the normal content. 2 have an N. If you see, we have 6.6 into 10 to the power 9 base pairs. But what about our gametes? Our gametes will be having only half of it. So 6.6, if you take half of it, it will be what? 3.3. Haploid content is 3.3, and over here it is 6.6. Now in between you have bacteriophage lambda with 48,502 base pairs and E. coli with 4.6 into 10 to the power 6 base pairs. Please remember this. This can come. Okay. Hi Vishesh. A very good evening, dear. Okay, so these are the things that you have to keep in mind. The highest is human beings. The lowest is bacteriophage 5x174 with the least. If at all you do not remember the numbers, at least remember high, who is having high, who is having the least. This is going to help you out. Now the second concept which is usually being asked is, please tell the DNA helix structure. This came in preparatory 1, right? So how do you explain the DNA helix structure? My dear children, your DNA is made up of what? Your DNA is made up of nucleotides. Okay, a nucleotide, it's not just made like this. You need some certain components. So over here you have those components. What is that? First thing you will need is pentose sugar. Do you have pentose sugar? Yes. Second thing you will need is nitrogenous base. The third thing that you need is what? A phosphate group. Three of them, when they combine together, I say it's a nucleotide. But suppose we are not combining them all of together. We are talking one by one. So let's talk about it. The pentose sugar that you are having right over here, in the case of DNA, you call this as what? Guys, you can also tell me the answer for this. The pentose sugar that you have over here, what do you call this? There are two types of sugars, deoxyribose sugar and ribose sugar. Deoxyribose sugar is there and ribose is there. So usually in ribose sugar, no, here on the second carbon, so this is the first carbon, this is second carbon, this is third carbon, this is the fourth carbon, and this guy is the fifth carbon out there. On the second carbon, you have an O. In what? In the ribose sugar. But over here, my dear children, in deoxyribose, this O is not there, which is why you call this as deoxy. Okay, deoxy. So deoxyribose sugar will be there in the case of DNA, and here you will be having nitrogenous bases. And your nitrogenous bases are how many types? You have purines and pyrimidines. Purines are adenine and guanine, pyrimidines are cytosine and thymine. And in the case of RNA, they also have one more thing that is known as uracil. So they do not have thymine. Instead of thymine, they will be having what? Uracil. Bhumika, hello dear. Okay. Now, I already spoke about pentose sugar, deoxyribose in the case of DNA, ribose in the case of RNA. Now, phosphate group is also there. Now, RNA versus DNA, what is the difference between them? RNA has an additional OH at the 2 carbon of ribose. So, I told you, RNA will be having O over here. Deoxyribose does not have that. Okay. And this O makes the, RNA very, very reactive as well. Now talking about RNA, once more thing, RNA contains uracil instead of what? Thymine. So over here I told you there are multiple bases, pyrimidines, right? So instead of thymine, they will be having uracil. But who came first? RNA came first or DNA came first? It was RNA which came first. So first you had uracil. First thing that you had was uracil. Now onto this uracil, on the 5th carbon, methyl group was added, and that's how you got the thymine. And this is also a question. What is the other name for thymine? It is what? It is 5-methyl uracil. So on the 5th carbon, if you had a methyl group, you have 5-methyl uracil. So my dear children, 3 components of DNA, nucleotide, I hope it's clear, pentose sugar, and you have your nitrogenous base, and you have what? You have your phosphate group. Now, one very important thing that you have, and that is, when you exclude this guy, when phosphate is not there, just pentose sugar and nitrogenous base is there, you will call this as nucleoside. What do you call? Nucleoside. When all 3 guys are there, you will call this as what? Nucleotide. Over here, between the pentose sugar and the nitrogenous base, there is a bond. What do you call this bond as? You will call this as N-glycosidic bond. N-glycosidic bond between the sugar and the nitrogenous base. Now, you have one more bond over here between the pentose sugar and also the phosphate group. What do you call this bond as? You call this bond as phosphoester bond. Okay, over here, if you see the 5th carbon over here is bonded with the phosphate. You call this bond as phosphoester bond. Phosphoester bond. And now you need to remember one more very important thing, and that is how exactly is DNA going to be formed. So look at me. You have this sugar. This is your pentose sugar. On here you have your nitrogenous base. Here you have your phosphate group. Okay, the phosphate group is basically attached like this. So here you have the 5th carbon, 1st, 2nd, 3rd, 4th, 5th carbon is there. Onto the 5th carbon you have the phosphate group. This bond is known as phosphoester bond. Phosphoester bond. Now suppose there is another nucleotide at the top. Suppose there is another nucleotide at the top, the same thing is going to happen. So over here, 1, 2, 3, 4, 5th carbon will be there, and 5th carbon will be having phosphate group. Now this 3rd carbon will be also attaching to the phosphate group right over here, and that is how you form what? The DNA structure. Now you have 2 carbons, basically a carbon over here attached to phosphate. Here also carbon is attached to phosphate. This bond is also known as phosphoester bond. This is also known as phosphoester bond. Together, what do you call this as? Phosphodiester bond. Okay, phosphodiester bond. When you talk about both of these carbons, you call this phosphodiester bond. So always remember, phosphate and sugar, they form the backbone of DNA. It is a very important question. Phosphate and sugar forms the backbone of DNA. Got it? Yusuf Samarth, hello dear. Okay, now over here, if I talk about the structure directly, because I see whenever concept is being taught, you guys get worried. So let's see. what is this points that you need to write? DNA has two anti-parallel polynucleotide chain. One is from 5' to 3', other will be from 3' to 5', ulta, okay? With sugar phosphate backbone and bases projecting inwards. So if at all you see, bases are inside and the sugar phosphate bone is basically outside. One is in 5' to 3', other is in 3' to 5'. This is the first point that you need to write. Second thing, base pairing via hydrogen bond is there. Base pairing is there. The nitrogenous bases which are there, they have base pairs. That means adenine which is there, it base pairs with thymine, okay, and guanine which is there, it base pairs with cytosine. How many hydrogen bonds are there? Adenine and thymine just have 2. So T for 2, T for thymine, you can remember like that. Guanine and cytosine, G and C looks very, very similar. So I'll say that basically they have 3 bonds. Okay, they have 3 bonds, 3 hydrogen bonds. Now, here the DNA is having a right-handed helix. What type of helix? Right-handed helix. And also remember, if at all you consider from this portion to this portion, this is considered as one helix. Okay, what do you call this? This is considered as one helix. And the pitch of this helix is 3.4 nanometer. And suppose I am telling you that over here you have 10 base pairs. In this particular range, there are 10 base pairs. And in between each base pair, this particular gap that you have, it is of 0.334 nanometer. 0.34 nanometer. So what is the pitch of DNA? It is 3.4 nanometer. And what is basically the space between each base pairs? It is 0.34 nanometer per basis. And base pair stacking and hydrogen bonds provide what? Structural stability. These base pairs which are there, which are arranged together, and also the hydrogen bonds between them, they provide what? Structural stability to the DNA. Always remember, the backbone is sugar phosphate backbone, and the bases project inwards. So this is what the DNA helix structure. This was asked in preparatory 1. Probably it might be not asked, but be prepared. We do not know. Okay, and thymine is also known as 5-methyl uracil. Do not forget that. DNA is much more stable than RNA. Why? Because DNA does not have OH on its second carbon. RNA has it. Cool. Now we go question by question. First question is this. Name the methodologies used in human genome sequencing. So what was the methods which were used in human genome sequencing, guys? Any idea? Provide hot questions. So I am going to start off with the questions right now. And from this point onwards, it is going to be a very nice journey. So those of you who were there in preparatory 1, you guys know how we study, right? So are we ready? Okay, so I just gave you a brief of something. Let's go ahead. It's super important. And it goes like this. Listen to me carefully. How do you basically do human genome sequencing? How do you sequence the entire DNA? There were 2 methods. One is expressed sequence tag. This is first one, where basically there is DNA, and the DNA, when it is transcribed to mRNA, the mRNA will be having only the regions that is going to code for proteins. You know, DNA has regions that codes for proteins, and DNA region that does not code for anything as well. But now we are just going to code only the sequences which will be giving down proteins. This is known as expressed sequence tags. What do you call this? Expressed sequence tags. And now there is also one more method where you are not seeing what is getting expressed, what is not getting expressed. You are basically going to sequence the whole of it. This is known as whole genome sequencing. So sequence annotation or whole genome sequencing is where the entire genome, coding, non-coding things, everything is sequenced together. So these are the 2 methodologies that are used in human genome sequencing. This is a 2-marker question. This came in your model question paper 1. It's an important question. Okay, got it? Can I go ahead? It's an important question. Okay, going ahead with the next thing. Draw a label diagram of nucleosome. Now nucleosome structure is also super important. How do you draw it? Listen. The nucleosome is basically what? First of all, you need to know that. So let's see. Over here, my dear children, your DNA is 6.6 into 10 to the power 9 base pairs. So basically, it is 2 meters long. This 2 meters long DNA, can you put it inside the nucleus? It is going to be really tough. So you have to make sure that it is highly compact. How do you make it compact? With the help of nucleosome. And how are you going to do that? Let's see. So in the case of nucleosome, you have 1, 2, 3, 4, 5, 6, and in the back side you have 7, 8. 8 what? 8 histone proteins. 8 histone proteins will be there. And this histone proteins which are there, my dear children, are basically containing 2 very important amino acids which are positive. Okay, which are positive. It's Habiba. Ma'am, I scored 61 marks in first preparatory after seeing your lectures. Really, you are great, ma'am. Thank you so much. And now I am aiming for 65 plus. Thank you so much, Habiba. It means a lot. Hello, Ramesh. Ramesh, yes, Ramesh. Okay, dear. I'm so happy and so glad and so proud of you. Let's go ahead. So over here, my dear children, you have the histone proteins, and I told you there are 2 amino acids. What are the name of the 2 amino acids? Over here, you have arginine and lysine. Arginine and lysine, AL, all. You can remember like that, all. Okay, these 2 amino acids which are there, they are positive in nature. They are positive in nature, and our DNA is negative in nature. Okay, it is negatively charged. So positive, negative, what happens? They attract, they come together. Negative, negative, they are going to repel. So over here, you want to make it compact. So you need to make sure you have some positively charged amino acid, because if it is positively charged, your DNA is going to wrap around it, and your DNA wraps around it twice. How many times? Twice, my dear children. 2 times. If it wraps around two times, how much nucleotides are there in this particular wrap? There are almost 200 base pairs. Please remember, 200 base pairs of DNA are wrapped around that. And this histone which is there, I told you there are 8 of them. 8 of them, when they come together, you call this structure as octamer. What octamer? Histone octamer. And also you have one more histone protein right over here. You call this as H1 histone. What do you call this? H1 histone. Okay? Fine? Fine. Let's go ahead. So over here the picture looks like this. As I told you, the DNA is wrapped around the histone. One extra histone protein you have, you call it H1 histone. Over here 8 histone proteins come together. They are rich in 2 things, arginine and lysine. Okay, they are positively charged. DNA wraps around twice with 200 base pairs, and H1 protein between 2 nucleosomes. So this whole structure that you see is a nucleosome. Multiple nucleosomes will be there, and multiple nucleosomes, my dear children, are going to form what? Chromatin. Okay, multiple nucleosomes basically is nothing, your DNA wrapped around it, and you call this structure as chromatin. Chromatin, when it condenses more, you call this as chromosome. Okay? Ma'am, I scored only 31. Any tips? Watch this video. And ma'am, you're my favorite teacher. Thank you so much, Red Stormer. HD Ramesh, ma'am, what was your KCET, JE, Comet K second PUC result? Baba, I am a biology student. I did not need to write JE and Comet K. My second PUC result was super great. Okay. And KCET also, I didn't have to write it, okay? Because I am not from Karnataka, so I didn't have to write it. Fine. Now, let's go ahead with the next thing. That is the question number 3. Which sequences of bases transcribed from DNA are found both in hnRNA and mRNA? Now, listen, this is an important question. Which sequences are found in hnRNA and mRNA? Simple logic you put and you understand, okay? Your DNA which is there, you want the DNA to basically form proteins. But do you think it's a one-step process? No, Baba, it's not a one-step process. So over here, you need to undergo 2 very important steps. One is transcription. Where what are you going to form? You are going to form an RNA. And now this RNA, my dear children, is going to undergo translation, and then it is going to form proteins. Now, even if it seems similar, very easy, but it is not that simple. The first time the DNA is going to make up RNA, it is not mRNA. It makes an RNA, and you call this RNA as. Ayyo, where is it? You call this RNA as hnRNA. HN stands for heterogeneous nuclear RNA. Okay, hnRNA. Now this hnRNA is just like the DNA. Whatever information is there in the DNA, exactly all of it is copied in this hnRNA. That means there will be coding regions, okay, and there will be non-coding regions. The coding regions which actually give rise to proteins, my dear children, you call them as exons. And the non-coding regions which comes in between, you call this as introns. What do you call them? Introns. Now, do you think we need introns, the non-coding ones? We want to make the protein, and in between that, do you need the introns? No, Baba. We just need the exons. So basically, there should be a process where the introns are being cut down. This process is known as splicing. Okay, this process is known as splicing. So splicing is being done. Now you just have the exons, fresh piece for making proteins. This thing which is there, it is known as the mRNA. And now this mRNA is going to undergo translation. So here the question is, what is common between hnRNA and mRNA? The only thing that is common is what? This thing, and that is exons. And that would be what? exons. There is only one thing, and that is what? Exons. Got it? I hope that is clear. So over here, if at all you see the picture, it will be more clear to you. If at all it was not clear. So exons are there, Baba. They code for proteins. Here in between, you have dirty introns. We do not need them. So we cut them out with the help of splicing. This was an hnRNA, and now you are getting what? mRNA after splicing. Okay, and now it is going to form proteins. Now remember, after transcription, there are some post-transcriptional modification. After transcription, after making the RNA, there are some modification. This is one type of modification, which is known as splicing. There is another type of modification where basically it is known as capping, where in the mRNA at the 5' end, you are going to add guanine group. You are going to add guanine group. Why? Because you want to make this particular side safe. Similarly, at the 3' end, you make it undergo tailing. Tailing means what? You are going to add adenine group. Okay, you add adenine group. You keep on adding it. Why? Because now this particular mRNA has to make protein. And do you think it can make protein inside the nucleus? No. It will move outside the nucleus. It will go to the cytoplasm. In the cytoplasm, there are so many villains to cut it off. So basically, to be safe, you have capping and tailing, and also splicing be done. I hope it's clear. Ma'am, tomorrow is my JEE exam. Can you wish me good luck? Wishing you good luck. I hope you basically ace the examination and come. Okay? Fine. Now, mention the levels where gene expression can be regulated in eukaryotes. My dear children, this is the next question that is most probable, and that is, how do you regulate gene expression in the case of eukaryotes? Any idea? Eukaryotes and prokaryotes are different. We are eukaryotes. We are eukaryotes. Basically, in eukaryotes, the cell will be having a proper nucleus, having nuclear membrane, everything out there, and cell membrane bound organelles, lot of things will be there. But in this particular case, in the case of prokaryotes, Papa Guy does not have anything. Okay, they do not have nuclear membrane, do not have a proper nucleus. Everything is scattered in here itself. Okay, in the normal cell. Completely in the normal cell. So obviously there are going to be some changes in the case of eukaryotes and prokaryotes. And here you are asked, how is gene expression going to be regulated in the case of eukaryotes? As in, when eukaryotes have all of this thing. Okay, just a second. Shakuntala, I hate biology. It's okay, dear. It's okay to hate a subject. Okay, it's okay to hate a subject, but if it gives marks, please study. Okay, so now we are in eukaryotes and we are seeing how does the gene regulation works. So basically it's asking you, how do we regulate that this particular gene makes this particular protein? If I do not want the protein to be made, at what all levels can it be regulated? So let's see. So this is a cell, and here you have the nucleus and here you have the cytoplasm. Okay, here is the nucleus and here is the cytoplasm. You have the DNA right there. DNA has to undergo transcription to form mRNA. This particular process can be stopped. First level where gene can be regulated is that basically at the transcriptional level. Second thing, this mRNA which is there, it has to move outside because I told you translation happens outside. So the next place where it can be regulated is what? At the transport level. Okay, transport of mRNA can be regulated. Now suppose it reached outside. The next process is basically what? Translation. It is what? Translation. So at this level also it can be stopped. I can stop the process of translation as well. So three things already you know, and I already spoke about one very important thing, that is processing. Remember, hnRNA to mRNA, that can also be stopped over here. So at transcription level, at processing level, at transport level, at translation level, gene regulation can be done in the case of eukaryotes. But in the case of prokaryote, it is not like that because everything is happening in the same region. You cannot regulate it so many places. Okay? Fine. Let's go ahead. So in eukaryotes, the regulation could be exerted at transcriptional level, processing level, and transport of mRNA from the nucleus to the cytoplasm, and also translation level. This is the model question paper question of the previous year. So this question is also important. Two more question. Okay? Guys, apart from Shakuntala who is hating biology, are there anyone else who is understanding it? Is everything good? Is everything good to go? Can we go ahead with the next question? Children, give me a thumbs up if at all everything is good and we can go ahead with the next question. You know what? You guys are amazing. I know you might be tensed, like watching this video is going to help you or not, but let me tell you, it is definitely going to help you in some ways for sure, because I will be covering the important chapters. So at least you can be rest assured. You have time. I am here with you. I will be making sure that all your doubts are cleared. Okay? Okay, fine. Let's go ahead then. The next question that usually comes up is, what is the role of sigma factor and rho factor in transcription in the case of prokaryotes? So in the case of prokaryotes, my dear children, transcription is basically DNA to RNA, right? Who does it? It is done by RNA polymerase. Now this RNA polymerase guy which is there, no, RNA polymerase is like lonely person. It cannot do anything alone. It is dependent on something. So suppose it wants to start off transcription in the case of prokaryotes. So it needs a factor, and that factor is sigma factor. What factor? Sigma factor. So basically RNA polymerase is shy to come and attach on the DNA, so it needs sigma factor. S for shy, it's for sigma. So for initiation of the process, always remember you need sigma factor. This sigma factor plus the RNA polymerase goes and binds on the promoter region of the transcription unit, and that's how the whole process goes on. Now when it reaches the end, you see an RNA is being formed. When it reaches the end, my dear children, the RNA polymerase is starting to cry. It is crying. You call it as Rona in Hindi, right? So rho factor comes up over here and say that, bro, it's okay, chill Madi, it's okay. You just leave the RNA, and the RNA is also formed, and the rho factor also go. So basically for the termination process, you need what? Rho factor. Always remember sigma factor in the starting, rho factor in terminating. This is a very important point. Okay? Shalom, hi dear, hello Shalom. And young Thor, ma'am, please make important 2-3 marker questions, ma'am. So I had made a video on fixed questions. Okay, fixed questions. If at all you just want to refer to questions, you can go watch that video as well. So it was a previous video that I did, but yes, you can. I'll show you the video again. So listen. So over here, where is it? Yeah. Short PU2 fixed question. So the questions are not going to change. It is going to be the same in preparatory and this and the previous preparatory also, right? In the boards also. So the fixed question video has been done, as you can see over here. It is going to play or what? No. So if at all you go roll down a bit, you will be able to see this fixed question video. Go watch it, you will get the important questions. And now if you want the solution, my dear children, I had done a live, and apart from that live contained all the important chapters. Apart from the live, I recorded a video of the chapter starting from microbes and human welfare to what? To biodiversity and conservation. And those chapters is covered over here in Biology Preparatory 70 and 70 part 2 video. So you can go watch that as well if at all you need any help. Okay, fine, done. Can I come over here back? Yes. So we were right over here. How do you write this answer? So sigma factor binds to RNA polymerase, guiding it to the promoter region for transcription initiation. It detaches after initiation, as I told you. Next thing, facilitates. Rho factor facilitates termination by binding to RNA and unwinding the RNA DNA. When over here RNA is being formed, the RNA DNA complex is also being formed. So you need to unwind it. You need to separate it. Okay. So that also happens right over here with the help of rho factor. Got it? Let's go ahead with the next question. Write the difference between euchromatin and heterochromatin. Name the virus used in Hershey and Chase experiment. This is for you guys. You can tell me the answer for this. Okay, you can tell me the answer for this. And A part, write the difference between euchromatin and heterochromatin. My dear children, Shalom says, I am so cute. Is it? You guys are also so cute, my dear children. So listen to me. Over here, basically what happens, I told you nucleosome is formed. Nucleosome is what? Basically a gold round thing on which your DNA is wrapped. Your DNA is wrapped because you want to make it compact, because you want to fit it inside the nucleus. So when you do this, the structure which is going to be formed, the bead on a string-like structure. Bead on a string-like structure is going to be known as what? Chromatin. What do you call it? Chromatin. There are two types of chromatin. One is euchromatin, other one is heterochromatin. You are asked the difference between euchromatin and heterochromatin. This is an important question. So how do you write the answer for this? So listen to me carefully. Euchromatin, as you can see, it is loosely packed chromatin. Heterochromatin, they are very stacked up each other. So when this chromatin is loosely packed, basically your DNA is loosely packed. It is having space such that any enzyme can come over here, such as RNA polymerase, and can undergo transcription. You can make an RNA out of this DNA also. So we say euchromatin is transcriptionally active because RNA polymerase can come over here and do the process of transcription. What about heterochromatin? It is not transcriptionally active because you do not have any space for the RNA polymerase to come and attach. So this is transcriptionally inactive. This one is transcriptionally active. Now you tell me, if I put a stain on both of them, which one will be having darker stain? Obviously this one, this does not have any space, so that will be too dark in color, whereas this will be staining up lightly. So this will be having lighter stain. These are the two differences that you need to write. In a typical nucleus, some loosely coiled regions of chromatin with light stain and transcriptionally active is there, and that is known as euchromatin. On the other hand, the chromatin that is more densely packed and stains what? Stains dark, and transcriptionally also it is inactive. It is known as heterochromatin. Now over here you are asked the second part of the question. Anyone? Can you tell me the second part of the question? Anybody have question paper? That is the last step which is there in any examination. Sadly, I don't think anyone has the question paper, and even if they have, how can you be sure that it is the same question paper that will be coming in? So don't do that. Okay, study and go. Okay, name the virus used in Hershey and Chase experiment, guys. Can anyone tell me? The virus that is used in Hershey and Chase experiment. Come on, come on, come on. You guys know it. I want the answer from you. Listen. If I am the person who talks, I will be tired after some time. And then who will teach you? So you also have to talk to me. Then only it is going to be really nice. Hershey and Chase experiment, which virus was used? Any guesses? Hershey and Chase experiment, which virus was used? No guesses? No? Okay. In that particular case, I have to tell the answer. Since no one knows the answer, it is what? Bacteriophage. Bacteriophage, and you have seen the E. coli, right? And the E. coli. So bacteriophage goes and affects the E. coli. And bacteriophage is the virus. It is not the bacteria, it is the virus. It has a name like bacteriophage. So bacteriophage means it is eating up bacteria. That is why phage is being put down there. So it is a virus used in Hershey and Chase experiment. Okay, done, deal. Now let's go ahead. Now list the goals of Human Genome Project. This is also a question that can come. Yes, core, oh my god, that's a very complex name. Coreto surface. Core surface, I do not know, but yeah, you have given me the answer correct, and that is bacteriophage. Kudos. So list the goals of Human Genome Project. My dear children, in the previous preparatory question came, what are the organisms used in Human Genome Project, and everyone got scared, okay, because they didn't read the NCERT properly. But do not be worried, because over here I have made sure that you read everything which is important. So Human Genome Project, my dear children, if at all you're asked about the goal, write this. What is the goal? To identify approximately 20,000 to 25,000 genes in the human DNA. To identify at least these many number of genes in the human DNA, and also after identifying, you need to sequence this A, T, G, C. What is the sequence of it? You have to sequence it also. So this was the second goal. Determine the sequence of 3 billion chemical base pairs that make up what? Human DNA. And next thing, store this information. After getting the information, you have to store in what? Database. And after storing it in, you have to make sure you improve tools so that you can analyze this data also. So improve tools for data analysis. Next thing, transfer this related information to other sectors also, so that they can also make the best use of it. So that is the next thing. And the sixth thing is to address the ethical, legal and the social issues. Ethical, legal and the social issues, which is known as LC, that may arise from the project. So these were the goals of what? These were the goals of what? Your Human Genome Project. Yes, Shalam, it is bacteriophage. Awesome, dear. Okay. Now next thing which is given over here is this. The Human Genome Project was a 13-year-old project coordinated by the US Department of Energy and the National Institute of Health. Why I am telling you over here, you can expect any questions coming. So it was 13-year-old project, and it was conducted by US Department of Energy and also National Institute of Health, NIH, my dear children. Also one very important thing, and this is the question that came in preparatory 1. Have any other organism been sequenced apart from human being? Are there any other organisms that have been sequenced completely? Yes. What is that? Such as bacteria, yeast, okay, Caenorhabditis elegans, a free-living non-pathogenic nematode, okay, and also Drosophila, that is fruit fly, and plants such as rice and Arabidopsis have also been sequenced. Ma'am, do we need to remember the names? Yes. This came as a 2-marker question in preparatory 1. Okay, so bacteria, yeast, Caenorhabditis elegans, and then you have Drosophila, you have plants such as rice and Arabidopsis. These were the things which were sequenced apart from this. Okay, Sneha, ma'am, MCQs, dear, I was planning not to cover MCQs over here since it might take a lot of time. That is the reason I was not covering it up. If you guys want MCQs, I have to open another PDF. I can do that, but then you will be missing out on the 2 long mark questions. So the choice is yours. So DNA is genetic material in both prokaryotes and eukaryotes, yet the packaging process in their cells is not the same. Explain. This is your MQP, model question paper question. Okay, so it is said that DNA is the definite genetic material in prokaryotes and eukaryotes, but packaging is not the same. Why, Baba? It's not the same because you saw in in E. coli, the genome size, it was very different from us. Okay, E. coli has very little genome size, whereas us, my dear children, has very much. So their genome size is 4.6 into 10 to the power 6 base pairs. Ours is 6.6 into 10 to the power 9 base pairs. So genome size is different. That is one very important thing. The next very important question that comes up is that basically, do you have nucleus? If you have nucleus, packaging will be different. You would have to fit things in. This is why you will need nucleosomes. But if there is no nucleus, then do you need nucleosomes? No, Baba. So that is how in two ways prokaryotes and eukaryotes packaging is going to be different. The first one is in prokaryotes DNA is naked. DNA is naked means basically it is in the cytoplasm, in the normal place which is there. It is circular and present in the nucleoid region, a region which is there. That's all. Without any histone proteins. There are no histone proteins because there is no wrapping required. Because of small genome and absence of a nucleus. This is a reason. Now over here on the other hand, in the case of eukaryotes, DNA is associated with histones and basically they form histone octamer, there is nucleosome, their genome size is also very big, which is why the packaging is going to be different. If you write this, it's going to be good. Ma'am, for NEET, do you prepare for us? I used to, definitely the previous year. This year, with this lots of schedule coming up, I'm not pretty sure, but I'll definitely try. Okay? Okay, now going ahead with the next question that we have is explain Griffith's experiment. What was Griffith's experiment? Griffith's experiment was transforming principle experiment. This is a usual question. Okay, so how do you do this? So Griffith was a guy who was worried like what is the genetic material? What is the genetic material? He wanted to find that out. And what did he do to find the genetic material? Basically, he did an experiment. So for that, he used a very important bacteria. You need to remember its name. The name of this bacteria is Streptococcus pneumoniae. Streptococcus pneumoniae. And this Streptococcus pneumoniae had two strains. Okay, two strains. One was R strain and the other one was S strain. So R strain, R stands for rough strain. Rough means, ma'am, what is happening over here? I am going to tell you that. S strain means smooth strain. Ma'am, why is it smooth? Did it apply any moisturizer? The reason is that it had an extra polysaccharide covering. And this extra polysaccharide covering which was there, my dear children, made this strain virulent. If at all any organism got infected by this strain, they used to die. They used to basically die. So we not... Yes, rat is equals to Puchu. You remember Puchu? Oh my God. Oh, so sweet. Okay, Sanjana, hi dear. Hi. All of you, every one of you, please share this video with all of your friends who are also preparing for biology examination. This is going to be super important for them. Okay. Okay. Works. So over here, what is happening? What was I saying? Yeah. So smooth strain is there and R strain is there. So R strain is non-virulent, does not cause any trouble, but S strain is the virulent strain. Now suppose you have the mice, that is Puchu. Okay, this is the Puchu mice that we are having. Over here, we are going to insert the live R strain. When the live R strain is there, Puchu is not going to die. Puchu is going to live because it is the non-virulent strain that we are putting. Okay, understood. Now, when he put the live S strain, Puchu died. Okay, Puchu died. Why? Because it was the virulent strain. Understandable. We knew this. But now he did some changes. He heat killed or basically treated this with heat. The live S strain was treated with heat. So it was heat killed now. And when he put the heat killed strain into the Puchu, Puchu lived. Oh, that was a miracle. This was not expected. So that means because of heat, something was getting changed, which is why Puchu was living and this strain was not effective out there. So now he thought that this and this, anyway, both of them are not killing Puchu. So why not mix both of them and put it in the mice and see what happens? Now Griffith, my dear children, tried this up and you know what? Sadly, Puchu died. And not just that, in Puchu's body, live S strain was found. He never put live S strain. From where did live S strain come from? So that means this guy, live R strain, was getting transformed into S strain because something might have passed from here to here. Something might have passed from here to here. And what was that? That was the question. And even Griffith could not find the answer for this. That's why he said that it is a transforming principle that is getting transferred. But I do not know the answer for this. You guys do the experiment. And then, my dear children, came into picture who? Avery, MacLeod and McCarthy. Avery, MacLeod, McCarthy came into the picture, and they were in search of what? The genetic material. Was it DNA? Was it RNA? Was it protein? They tried to find it out. How did they try to find it out? Listen to me carefully. So over here, when this experiment was being done, so they did three things. First of all, they treated the heat-killed S strain with some RNAs. When you treat it with RNAs, all the RNA is going to get degraded. If RNA is going to get degraded, he wanted to check whether it is making the changes still happening, the change is still happening or not. So he treated with RNAs, and then he mixed both of them and he gave it to Puchu. Puchu still died. That means nothing important was getting degraded. The thing that was causing transformation, it was still there. It was still there, so it was not RNA. Now, the next thing that he used was protease. So now when he put protease into heat-killed S strain, all the proteins are going to be degraded. But still, when they mixed both of them and put inside Puchu, Puchu still died. That means protein was also not something which was causing trouble, because anyway, protein is all degraded now. And if there's no protein, how can this transformation happen? Next thing that he did was, basically he treated it with DNAs. And when they treated it with DNAs, it will be killing or degrading every DNA particle out there. Now, if DNA is not there, he saw that basically transformation was not taking place, and Puchu, my dear children, lived. Puchu was living. That means DNA is something that had been the genetic material. But you know what? There was a problem. There was a problem, and the problem was Avery, MacLeod, McCarthy were not accepted. People said that we need one more proof to finally agree that DNA is the genetic material, and that's when came Hershey and Chase. So Hershey and Chase did their experiment on DNA, finding the genetic material, and they got to understand that DNA is definitely the genetic material. That was the experiment. So the last experiment and the important experiment, go through Hershey and Chase experiment. Okay, it is important. Write it somewhere. Hershey and Chase. Bacteriophage was used. What was the other thing? If you have a doubt, you can tell me. You can ask me, do you want me to explain Hershey and Chase experiment? If yes, comment it down right now. I'll explain. If not, then I'll just move on. Okay? So please know about it. Sneha, when is your birthday, ma'am? My birthday is on 6th of October. So it is yet to come the next year. So Griffith experiment, my dear children, you have to write like this, that Frederick Griffith conducted his experiment. He used Streptococcus pneumoniae. This in bacteria's name is super important. S strain, R strain was there. S strain was virulent. R strain was non-virulent. And then he put inside the mice, this happened, this happened, this happened. And he got that. He concluded that some heritable material or transforming principle was getting transferred from S strain to R strain. That's all. No, ma'am. Yes, ma'am. So there are 2 yes and 1 no. Ooh. That's a tough competition. Okay. So I'll explain it to you, okay? I'll explain it to you. Anyway, we are here itself. So I can, and this is important also. Do I have the picture? No, I do not have the picture. So listen to me carefully, okay? Most important question. Sanjay Shetty, these are the most important question. In biology, questions can come from anywhere. I have made a video on fixed questions. If at all you want to watch it, go watch it and solve all the questions. But okay. So over here, bacteriophage, my dear children, was infecting E. coli. So this is the E. coli that we are having. We have 2 scenarios. There is one bacteriophage. Okay, and there is another bacteriophage. These 2 are basically viruses. Okay, this bacteriophage which was there, it was tagged with S35. S35, you can remember 5 looks like S, so 35 is with this. And this bacteriophage was tagged with P32. What? P32. So over here you need to remember one thing. Sulfur is a compound that we find in proteins, and the capsid is something that is made up of proteins. So sulfur is going to combine with the capsid, that is the outermost layer that you see. Whereas phosphorus, which is there, it is a component of your genetic material. Your DNA is made up of phosphorus, right? So this particular phosphorus is going to get inside your genetic material. Fine. Now both of this bacteriophage were attacking on the E. coli. They did the attack. It was done. After that, blending was being done. Blending means basically all of them were put inside some chamber and all of them were separated. So after the infection, so listen to me. Bacteriophage, when it infects, it basically transfers the genetic material inside. It basically transfers the genetic material inside. But over here, genetic material was not tagged. Over here, genetic material was tagged because it was made up of phosphorus, right? Now, people were not able to see it. Wait. Okay, listen to me carefully. Now what happened? Blending happened. You basically made everything separated and then you centrifuged it. You centrifuged it at high speed. So everything separated, and when everything was being separated, my dear children, it was found that there was no radioactivity seen over here in the pellet. So in the pellet is where the cells will be there. The pellet is the place where the cells will be there. And then you have supernatant, the extra fluid out there. Here radioactivity was there. Radioactivity was there. And the radioactive substance over here was protein. If the protein was in the supernatant, that means it was not transferred. It was not transferred. It should have been here, right? But on the other hand, if in this particular chamber, if at all you saw, my dear children, in the pellets itself, in this pellet itself, radioactivity was found. That means basically what? The DNA is the genetic material. DNA was getting transferred. So DNA is the genetic material was understood there. Whereas over here there was no radioactivity in the supernatant. In the pellet it was there, and the pellet you basically transferred. In the pellet if it was there, it was the DNA. So that is how things worked. Okay, things work. This is what Hershey and Chase experiment. He used bacteriophage, they used bacteriophage and E. coli. Done. Now we are going with the next question, question number 10. So those of you who wanted explanation, you got it over here, and please read the explanation from your NCERT also. Salam, advance happy birthday ma'am. Okay, thank you so much. Okay, fine. Let's go ahead then with the next question. State the arrangement of different genes of lactose operon in E. coli. Now this question is important. And explain the role of lactose in regulation of Lac Operon. So listen to me very carefully. Okay. So over here, if at all you see this picture, everything is going to be clear. So you have two genes. Lactose operon or Lac Operon is found in the case of E. coli. Where do you find it? In the bacteria. Bacteria such as E. coli. Okay. So this Lac Operon is there. You have two genes. One is this gene segment until here, and this is the next gene segment out over here. Okay. This gene is known as regulatory gene. Regulatory gene will have its own promoter. These genes which are there, they also have their own promoter. Okay. This regulatory gene is going to regulate something by doing some negative activity. Basically, they are going to produce a repressor protein. What are they producing? Repressor protein over here. The I gene gives repressor protein. Now, on the other hand, if at all you look at this, there is ZYA gene. The Z gene which is there, if at all transcription happens, it produces beta-galactosidase. The Y gene produces permease, and the A gene produces trans-acetylase. Okay, trans-acetylase. These are the three things which are being produced. So basically what happens is that beta-galactosidase is something that will be breaking down lactose to glucose and galactose. Lactose to glucose and galactose. And basically, only if lactose is there, this should be there. If lactose is there, there is nothing to be broken down, then why do you think transcription will be happening? So at this particular point of time, when we do not need it, do you think transcription should happen? No. We should stop the transcription. And how can that happen? With the help of this repressor protein. So this repressor protein goes and binds on the operator region of this. Now suppose our RNA polymerase came and attached on the promoter region and it wants to transcribe. Will it be able to transcribe? No, Baba, it won't be able to even move forward. It won't be able to move forward. Transcription is not going to happen. So that is the role of repressor protein. Now suppose in this particular medium, when the E. coli is growing, I am providing lactose. Now lactose is there. Now I need all these enzymes. Now I would need all this enzyme. Beta-galactosidase to break down lactose and to provide energy. Permease to increase the permeability of the cell membranes so that more lactose can come inside. And trans-acetylase to basically deal with everything in between. Okay, trans-acetylase deals with all the toxic substances that comes in between. Yes, Neha, tell me. Thank you so much. You're most welcome, dear. Yes. Okay, so this is happening out there. Now, the thing that is there is, I want transcription to happen because lactose is there in the medium. So this lactose slowly gets inside the cell membrane. Okay, gets inside the E. coli. And when this lactose is there, it goes and binds on the repressor. It goes and binds on the repressor. Now, do you think repressor will be available to go and bind on the operator? No, Baba. It cannot go and bind on the operator, which is why operator region is free and the promoter region also free. RNA polymerase will come and transcription is going to happen. In this particular condition, you say it is switched on. Transcription is what happening. This is known as switched on condition. And over here, who is inducing it? Who is making it happen? It was nothing but your lactose or allolactose. Okay, lactose or allolactose, basically they are going to act like inducers. They are going to help in inducing, and the whole type of different things are going to be formed. This is switched on condition and this is switched off. You need to remember the name of inducer. It is lactose or allolactose. You need to remember the name of enzyme, ZYA, beta galactosidase, Y is for permease, A is for acetylase, trans-acetylase. Okay, so ZIA is something that you can remember. Okay. Ma'am, can you explain the Lac Operon? Here it is the Lac Operon that I am explaining. Ananda, hi dear. Very big hello. Is it clear? Guys, Lac Operon is clear? So listen, over here if at all you see this, there is promoter, there is I gene. I gene is regulatory gene. And there is promoter, there is operator region, and the structural gene which is ZYA. Okay, now over here if we see role of lactose in regulation of Lac Operon. The repressor protein binds to the operator. Usually it binds to the operator. This blocks RNA polymerase, so structural genes are not transcribed. ZYA is not going to be transcribed. In the presence of lactose, what happens? Lactose or allolactose acts as an inducer. It binds with the repressor protein. Now the repressor cannot bind to the operator. If it is not binded to the operator, RNA polymerase transcribes ZYA and the whole process is going to happen. The lactose switches on the Lac Operon. So if lactose is not there, things are going to be difficult. Got it? Fine. Got it? Clear? Can I go ahead? Okay, ma'am. Fine. Thank you, thank you, thank you. So someone is showering hearts. Okay, let's go ahead to the next question. So over here we have the next question, important question. Given below are sequences of nucleotides in a particular mRNA and amino acids coded by it. Write the properties of genetic code. Properties of genetic code, yet another important question in this particular chapter. How do you write the properties of genetic code? Now let me know. Over here, see. 5' AUG, UU, UUC, GA, it goes on. Okay. Now over here, if at all you see, AUG is coding for methionine. What does that mean? AUG is the initiator codon. This is one feature of genetic code. AUG acts like initiator codon and also it codes for an amino acid, that is methionine. So AUG has what? Dual function, baba. It has dual function. Now what about UUU? UUU, UUC are coding for the same amino acid, that is phenylalanine. So two codons for one amino acid, two codons or multiple codons for one amino acid. This is known as degenerate features of codon. Multiple codons can code for the same amino acid. Same amino acid. Okay. Now over here, one more feature, if at all you see, all the codons are triplet in nature. They are all triplet. Three groups are there. And do you see any comma, full stop? No, they are all read in contiguous fashion. Continuous fashion is what you mean. Okay. Apart from that, if at all you see, one codon codes for only one amino acid. There is no cheating over here. This is known as unambiguous nature of codon. This is known as unambiguous nature of codon. And if at all you see, GUG, until here it is coding for valine. UAA does not code for anything. So UAA is what? It is a stop codon. We have three stop codons and the names goes like this. I'm going to talk about that. Here I have all the functions. Remember the name of the stop codon. One is UA, one is UGA and one is UAG. Okay. UA, UGA and UAG. So these are the three stop codons that you need to remember. So what are the features of the genetic code? The first feature is codon is triplet. Okay, three codons are there. AUG has dual function, second feature. Third function, some amino acid are coded by more than one codon. This is known as degenerate feature. And the codon is read in mRNA in a contiguous fashion. Next feature. Now UAA, UAG, UGA are what? Terminator or stop codons. And last feature which is there, which you need to remember, exactly Shalom, they are what? Universal. And this is an important MCQ question. They can ask you that this particular codon codes for this amino acid in human beings and it codes for the same amino acid in bacteria also. What feature does it show? It is universal. Everywhere the amino acid, the codons are going to code for the same amino acid, whether it's a bacteria, whether it's human being, everything. It is known as universal feature. Okay. And here you have exceptions. Please remember the exemptions also. This is going to help you well. What are the exceptions? So exceptions is found in mitochondrial DNA. So in mitochondria also you have codons, you have DNA. So in mitochondria mitochondrial DNA, it's an exception. Also in some protozoans, it's an exception. Rest everywhere, it's same. It's comma-less, exactly, Shalom. Got it? So these are the features of genetic code that you need to remember. Cool? Let's go ahead. Now, question number 12 is describe the steps involved in DNA fingerprinting technique. But before I go ahead, every one of you, have you understood what I'm teaching? I know I'm going fast, but if I do not go fast, you basically would not have time. But you should understand also. That is also one very important thing. So are you understanding? And if at all you have not clicked on the like button, click on the like button right now and share it with your friends and subscribe to the channel. Okay, so I know the time is pretty much less. Tomorrow you have your examination, which is why when I am a bit sad. The previous time I came, it was basically you had 2 days for your preparation, right? So I was a bit happy that I could explain you properly, nicely, but now it's Shahista. Yes, thank you Ananda, thank you Sanjana, thank you. Shahista is your name, Shahista Shreen. Okay. Okay, I'll call it like that then. Shreen, can I call you Shahista Shreen? Okay, anyways. So, describe the steps involved in DNA fingerprinting technique. How do you describe the steps involved in DNA fingerprinting technique? It's simple. Suppose I did a crime, okay? I did a crime and in the crime spot, my dear children, in the crime spot, my blood stain was there. But I said that basically Ananda did the crime, okay? I said that Ananda did the crime. Now, police came and police checked. Police came and police checked. And when they found that, is that there was some blood. There was some blood. Now, this particular blood was taken and checked and they got a DNA. Now, this DNA was coded. This DNA was basically coded, and after coding, basically, they made a complementary strand of the DNA. They made a complementary strand of this particular DNA and tagged it with some radioactive probe. This is what you call this. This is what you call as what? A probe. You call this structure as a probe. So you got the DNA, you made a complementary pair of it, and then you tagged it with radioactive probe. Now I'm going to tell you what you're going to do with this. Wait for a second. Now, my dear children, we'll go to the process of DNA fingerprinting. So me and Ananda, both of them were there, okay, and we were the suspect. So both of us, okay, so both of our DNA was being taken. Both of our DNA was being taken. So it was being put like this. Okay, then both of our DNA was cut. So isolation of DNA is done. Now restriction, with the help of restriction enzyme, the DNA was being cut into smaller pieces. So that was also done. Second step also done. Now after being cut, we make sure we make it run through gel electrophoresis chamber. So that is the next step, where with the help of gel electrophoresis, you are basically going to make this strands run on the chamber. So this strands, my dear children, were run. So I will change the color over here. So this is me and this is Ananda. Okay, this is me and this is Ananda. So my DNA strands, my dear children, they separated like this. And Ananda's DNA strand also separated like this. Okay, it was all clear. Fine. Now what? Now you cannot see this particular DNA strand in the gel electrophoresis chamber. You need to blot it. You need to blot it on a nitrocellulose sheet. So basically, with the help of nitrocellulose sheet, you blotted it and now it's on this particular sheet. Suppose this is a sheet that you are having. Oh, why is it moving like that? Okay, now you have blotted it onto a sheet. Now what? On the sheet also it's not visible to me. So now what I need to do is that basically I'll run the probe. I will run this particular complementary probe that I made before. And now when this complementary probe is being run, you know what happens? It will only go and attach to the regions where it is complementary. So this was the person's DNA who did the crime. You made a complementary strand to it. And I was the person who did that. So it will go and bind on my DNA strands like this. It will go and bind on my DNA strand and it will be showing up. And if at all you put autoradiography machine under the autoradiography machine, it will be showing up and it will be clear that I did the crime and Ananda was not the person who did the crime. Got it? This is your DNA fingerprinting technique. This is how you need to explain it. We all have certain regions which are similar, which is very unique. You do not have anything like me. Okay, we are different in certain cases. There are certain regions which are known as variable number of tandem repeats. Such as I say that in my body AGCT, in my DNA AGCT is repeated 14 times, but in yours it might be repeated only 11 times. We are unique in there, and with the small differences that we have, we can find out who did the crime. Got it? Okay. Fine. So over here, you have the first thing, isolation of DNA. That is the first process. DNA is extracted from the sample, and over here, what are the samples? You need to know that also. Blood, hair, saliva. In your blood, your erythrocyte is not being considered. This is an MCQ question. Write it somewhere. Erythrocyte is not going to be considered. Why? Erythrocyte means RBC. Baba, RBC is not being considered. Anyone, can you tell me the answer? Why? Why RBC cannot be taken? Suppose I have blood over here and I am saying I am taking the RBC for checking up. Do you think this RBC is of any use for me in DNA fingerprinting process? The hair is, the saliva is, because all of it has, my dear children, DNA. It also has DNA, but RBC does not have. Okay, how many chapters will be completed, Ananda Baba? Molecular basis of inheritance, principles of inheritance and variation, sexual reproduction in flowering plants, human health and diseases, and evolution. These are the chapters that I am planning to cover over here itself. Rest, you have the recorded videos, please watch it. You have the previous live also that I took. You can watch that also. Okay? Okay. You are my friend's crush. Oh my God. I am older to your friend. Please tell them. Okay. Okay, he is also watching the live. I hope your friend is studying biology with me. Okay, next one is what? Digestion by restriction enzyme. You are going to cut it with the restriction enzyme. Third one, separation by gel electrophoresis. You are going to separate it with the help of gel electrophoresis. Now you are going to do blotting. This is known as southern blotting. After that, you are going to hybridize with DNA probes. I told you, now the DNA will go match. Hybridization is required over here. And then you are going to visualize under UV rays or UV light. Okay, and then it has been done. That's done. DNA fingerprinting is done. Everyone who thinks DNA fingerprinting is done, give me a thumbs up. And also, the reason why erythrocytes are not considered for DNA fingerprinting is that your RBC or erythrocyte, your RBC or erythrocyte does not have nucleus. Do you know that? It does not have nucleus. If no nucleus, no DNA. Why it does not have nucleus? Because it said that I need space for storing hemoglobin. My role is to transfer oxygen. So please not give me what do you call nucleus. So it had nucleus before, but later on the nucleus is being removed when it gets matured. Okay, so you should know that. Not removed, it goes off. Okay. Why do not take? Okay, Ishwarya, I hope I answered your question. Okay, thank you guys for giving me thumbs up. Thank you. Lots of love to you guys. Okay, next question that we have, since your question is clear, is construct the following. Construct a complete transcription unit with promoter and terminator on the basis of hypothetical template strand given below. Now this question again is important. So over here, a hypothetical strand is given to you. You are being asked, make a full transcription unit, full proof transcription unit. How will you make that? How are you going to make that? So listen to me carefully. First of all, you need to know what direction is this. So whenever the arrow is in this direction, understand that this is basically the 3' end and this is going to be the 5' end. This is 5' end, this is 3' end. And this is what? Your strand. Your strand. Now you have to make a complementary strand to it if at all you want to make a transcription unit. Transcription unit means what? Basically your DNA, double-stranded DNA, where you have the promoter, where you have the terminator region. This whole thing becomes a transcription unit. Now if it's one strand is in 3' to 5' direction, you have to make another complementary strand in 5' to 3' direction. Got it? And then you have to do one more thing. Write the RNA strand transcribed from this. This is the DNA. You have to transcribe it and form the RNA. How do you do it, ma'am? So listen to me carefully. So this is how we do it. So, just a second. Yeah. So this is a strand given to us, right? 3', 5' direction, we wrote it. Now over here, we have to write the complementary. A base pairs with T, okay? T base pairs with A, G base pairs with C, and it goes on. You made double-stranded DNA. This strand, my dear children, always remember you have to mention that this is in 5' to 3'. Now, you guys tell me, RNA polymerase transcribes a new strand. Okay, transcribes RNA in which direction? In 5' to 3' direction. It makes a new strand in 5'. DNA polymerase, RNA polymerase, mRNA translation, everything happens from 5' to 3'. So over here, my dear children, if this is a strand that we have from 3' to 5', complementary to it, I'll make a 5' to 3' strand. My RNA polymerase is going to make a strand on the basis of this strand. So what do you call this strand which is going to act like a template? You will call this as a template strand. This is the template strand which is going to undergo transcription, whereas the other one which is not going to code for anything, you call this as a coding strand. I know the name is very much deceptive, but please do not fall into the trap. Coding strand is not going to undergo transcription. Template strand is too. The one in 3' to 5' direction is going to undergo what? Transcription. This is known as template strand. Okay, and over here is the promoter region, because here is where RNA polymerase will come and attach and transcription is going to be done. There you have the terminator region. Okay, and you say the position of promoter and terminator region according to coding strand. You say it according to coding strand. That means you say promoter is located at the 5' end of the coding strand. You need to give some respect to the coding strand also, right? Because anyway it is not doing transcription, but you need to consider it. That's why you say that. Similarly, terminator is located on the 3' end of the coding strand. Now, when you are asked to make an RNA, okay, now when you are asked to make an RNA, how do you make an RNA? How do you transcribe an RNA? Simple. You do not need to do too much of work. You already made a complementary strand of this, right? Just replace T with U, because in the case of RNA, thymine is not there, uracil is there. Thymine is not there, uracil is there. So just replace it with U. So UACG, UACG, hey, it is not T, okay, it is going to be U. So in the case of RNA, you do not have T, do not go right and come T and say that Jasmine Mam said that. So it is going to be U. And please mention 5' to 3'. Done? Clear? Is this clear? Will you be able to make transcription in it? This is the transcription in it, and basically in the case of prokaryote, sigma factor comes and attaches with the RNA polymerase over here for initiation, and rho factor attaches for termination. I discussed about this in the initial part of this particular video. Is it clear? If it's clear, can you give me a thumbs up? Everyone, give me a thumbs up. Aisha, study NCERT. Exactly, we need to study NCERT. Very, very important. Clear, not clear, clear, not clear. Shall we go ahead? Ayyo. Okay. Thank you, Ananda. Okay. Fine. Now you have the next question, that is, list the salient features of human genome. So human genome project's goal we saw. Now we are going to see the salient features of human genome. Thank you, Sanjana. Okay, let's see what are the features. So you need to remember certain numbers, and I have marked the numbers in yellow. First is, the human genome contains how many base pairs? 3164.7 million base pairs. Remember, over here everything is in million, nothing is in billions. Okay, 3164.7 million base pairs are there in our human genome. Now, average gene consists of 3000. So average gene which is there, general gene which is there, which is going to code for proteins, it just contains 3000 bases. This can be a question. A question comes, HNRNA, general average size of HNRNA is what? So HNRNA is what? Just a gene, my dear children, transcribed version of DNA, right? So what is the average size? It is 3000 base pairs only. Don't go write something else and come. Okay, but you know what? It varies. The average size is this, but there is dystrophin, a protein which has 2.4 million base pairs. Again, it's million. 2.4 million base pairs. The average is 3000, but over here it is 2.4 million base pairs. Now, the total number of genes is estimated at 30,000. How much? 30,000. Much lower than the previous estimates of 80,000 to 1,40,000. Okay, so almost 99.9% nucleotide bases are exactly same in all the people like us, and we are just different in 1%. Now, one more thing, the functions are unknown for over 50% of the discovered genes. How much? For 50% of the discovered genes, we do not know their functions, and less than 2% of the genome actually codes for the protein. Okay, less than 2% of the genome is going to actually code for proteins also. So whatever we know, 50% of them, the function also we do not know, even though we have discovered them, and less than 2% of the genome actually, whole genome actually codes for protein. What about the next thing? What is the rest of the things which do not code for anything? You call them repeated sequences. They make up the large portion of the human genome. Repetitive sequences are stretches of DNA that are repeated multiple times. And they are repeated, they also vary from person to person. Okay, they also vary from person to person. So I say that it might help in understanding evolution, how it has happened and everything out there. Now, another information which is important is this. Chromosome number 1 has the most number of genes, and that is 2968. So 2968, here there is no 7, rest every number is there. 2968. And chromosome number Y, the chromosome number Y is found in the case of males, and males, my dear children, have the fewest number of genes, and that is 231. So please remember the number. This is also an MCQ question that I can see. Okay, got it? Got it? Next thing. Scientists have identified about 1.4 million locations where single base DNA differences occur. That means basically we are just different at single base pair differences, that is known as single nucleotide polymorphism. That also scientists have identified. So basically these are the things that you need to remember. Starting from this, 3164.7 million base pairs is the whole genome. Average size is 3000. Dystrophin has 2.4 million base pairs. The total number of genes is 30,000, and for 50% of the genes discovered, we do not know their function. Only less than 2% actually codes for proteins. Okay, rest of them is repetitive. Chromosome number 1 has the highest number of genes, 2968. Chromosome number Y has the least, and that is 231. And there are single nucleotide polymorphism also seen in a body. Okay. Hello, Vani Zohab. No, Baba, it is 12th grade. Today it's 12th grade over here. Hello, Shivanand. Hi, hi, hi. Okay, done. That question is done. Human Genome Project is done. Can you give me a thumbs up? If you give a thumbs up, it will be really, really nice, because I'll understand that who all understood the concept. And also, who all have not liked the video yet, please like it. Please go and like it. This is super important, right? Okay? Here, just a second. Done. Let's go. Thank you so much. Now, explain the process of translation in protein synthesis. What is translation? From mRNA, when we make protein, this process is known as translation. Now, that is also super important. Let me tell you why it is important. Okay, listen to me carefully. Or how it is happening. So, first thing that we need is mRNA. You need mRNA. And this mRNA strand, my dear children, is 5' to 3' direction. Is in 5' to 3' direction. This is a new strand that we have made, right? This mRNA strand will be having some codons. For example, A, U, G, and the list goes on. It goes on. Now, there should be someone to understand that this codon is coding for this particular amino acid. So, who is that person? It is the tRNA. And your tRNA, it looks like this. It looks like this. So, there is 5' end and there is 3' end. This is important. On to the 3' end, you have amino acid being joined. And this process requires a lot of ATP. ATP is required very much in this particular process. Okay. This process is known as amino acylation or charging of tRNA. What do you call this? Amino acylation, or you call this as charging of tRNA. AUG, which is the codon, you will have an anticodon over here. That means opposite codon. A bears pairs with U bears pairs with A, and G bears pairs with C. So you will have an anticodon group right over here, and this particular tRNA will also be attached to an amino acid. If AUG is the codon, the amino acid will be methionine. Will be what? Methionine. But this process is not going to be super simple. So the first thing that is going to happen in translation is charging of tRNA. That means this tRNA is going to attach to an amino acid. It requires a lot of ATP. This is the first process that happens. Later on, suppose the mRNA is there. Suppose the mRNA is there. This mRNA which is there, it is going to be bounded by ribosomal unit. You know, you might have heard about ribosome. There is larger subunit of ribosome and smaller subunit of ribosome. This is an organelle that you find in cytoplasm of the cell. From the nucleus, the mRNA came out to make the protein, and in the cytoplasm, it met the smaller subunit. So, smaller subunit came into the picture. Then your charged tRNA will be coming. So, this charging happened earlier. Now, this charged tRNA is going to come and bind over here. Later, the larger subunit is also going to bind, and now we say that it is ready for the process of translation. Okay, it is going to be ready for the process of translation. This is being done. Now what is going to happen? My dear children, slowly tRNA is going to come and attach over here. Again another tRNA is going to come and attach over here. And the ribosome is moving like this. So as you see it is moving, what is happening? Amino acids are getting joined. So now this guy is going to move away. And whole of this chain is going to go to this particular guy. Again next guy will be coming and whole will be going to the next guy. The same thing process goes on repeated, my dear children. And in between the amino acid you need to make a bond. This bond which is there between the amino acid, you call this bond as peptide bond. Ma'am, who makes the peptide bond? Over here in the ribosome. Ribosome is made up of what? Ribosomal RNA, rRNA, and also some important proteins. So some of these proteins are going to help in what? Peptide bond formation between two amino acids right over here itself. Okay, and then elongation is going to happen. Later on, you know, mRNA will be having a codon that is UAA, UGA or UAG, the stop codons. Onto this codons, no tRNA will come and attach because it is the stop codon. Rather, release factors, some factors known as release factors will come and attach, and that's when we say that the process has ended off. Okay, the process has ended off. So over here, if at all I want to write, I can write like this. First is activation or charging of amino acid, and that is being done by charging of tRNA, that is being done by this particular process. Okay, next is initiation. mRNA binds to the small subunit, and then the start codon is recognized by initiated tRNA, the first tRNA. Then what happens? The large subunit also comes, forming the whole complex. So initiation part is done. Now elongation. tRNA brings amino acid to the ribosome, matching codons on the mRNA through its anticodon. This thing happens. Now peptide bond forms between the adjacent amino acid. Okay, and this name of the enzyme you do not need to remember that. Just remember peptide bond is formed. But in your NCERT, one thing is being written, and please remember that thing. The ribosome also acts like a catalyst in bacteria. Ribosome which is there, it also acts like a catalyst. Catalyst is something that will be helping in the reaction. There might be certain proteins in the ribosome that will be helping in the reaction. And in your bacteria, there is 23 sRNA. 23 sRNA, ribosomal RNA, which is going to act like an enzyme, that is ribozyme, and help in peptide bond formation. Please remember, this is found in what? A bacteria. This helps in peptide bond formation. This is a part of your ribosome itself. Now termination is the last process. When a stop codon is reached, translation stop as the release factors is going to bind. Now one very important thing before I end up this, and that is UTR. This question came separately in a 2-marker question, that is what is untranslated regions or UTR and where are they found. So basically over here, if when you talk about an mRNA, before the 5' end and after the 3' end, you have some region which is not translated. These regions are known as UTR, untranslated regions, before 5' end and after 3' end. Why are they there? For basically providing stability to the mRNA, for helping in the process of initiation. Only when they are there, initiation is going to happen. Also, they help in protection of the mRNA. So 3 things remember. First is basically they help in efficient translation. Second thing, stability to the mRNA. Third thing, for smooth initiation as well as termination. You have this. Okay? Got it? What is the question? Ma'am, MCQ. So dear, as I said, MCQ I'm not planning to discuss over here in this particular live, because MCQ might require a lot of time. I'll request you to go watch the previous live which is there. So if at all you have seen, where is it? Yeah. If at all you see the previous live, where was the previous live? So here is the previous live. If I zoom in, what will happen? Let me see, okay? No, I cannot zoom that in. Just a second. So basically, if at all you see this live, it was pretty long live, but it was really useful, my dear children, because I saw a lot of children commenting down on it. Over here, I had spoken about the MCQs. I also gave the 2, 3, 5 marker questions of all the things that I have discussed over here. But in this particular live, I have taken only the 2, 3, 5 markers. Why? Because MCQs might require a lot of time. I will not be able to complete evolution chapter and all. So if you want that, you can tell me. Okay, so either way, you can go ahead. Okay? I'll recommend you take the PDF. I will be giving you the PDF, or you will find in the what you call WhatsApp group itself. If at all you have joined the WhatsApp group, you can go through the PDF, solve the questions as well. That way also works. So you can do that. Okay, if that helps, you can do that. I do not mind. I do not mind you not attending this live if at all you feel your concepts are really strong. Okay? Fine. This video is not for views. This video is for you guys to get marks. So if you think that the previous video had helped you, you can watch the video only. You can take the PDF from there only. You do not need to spend your time over here. Okay, but if at all you feel that you want me in front explaining your concept and asking doubts, you can be here. Cool? Okay. Next thing. So question number 16, which is there, DNA replication is said to be semi-conservative. And as I told you, just one more thing for those of you who came late, as I told you, so in the live I had completed, just a second, okay? In the live I had completed, I think, molecular basis of inheritance, sexual reproduction in flowering plants, human reproduction, principles of inheritance, and one more chapter was there, that and all I completed in the live itself, human health and diseases. Rest of the chapters which were there, for example, this thing, what do you call that as, what is the name of the chapter? Oh my god, from microbes in human welfare to the other one, to biodiversity and conservation, I put it in recorded format. Okay, I put it in recorded format. So if at all you want to see that, I can show you once more, just a second. Give me a quick little second, okay? So over here, as I said, this was for the chapters from principles of, from the first chapter to human health and diseases. All of this was covered in this particular live. You can see second PUC preparatory part 1 to 5 marker. Okay, this was covered and this was really helpful for children. Now, there was one more thing that I put in recorded format for the other set of chapters that was recorded. I think it was recorded, which is why it was not viewed much, but it's going to help you out. You can watch this part 2. Okay, here you have MCQ questions for the other chapters as well. Ma'am, on 28th my exam is there. Oh my god, all the best. All the very best. Okay, now I think we are on the last question of this particular chapter, and that is DNA replication is said to be semi-conservative. Describe the experimental proof by Michelson and Stahl. Let's see. Okay, let's see Michelson and Stahl experiment. So listen to me very carefully. What do you mean by semi-conservative mode of replication? Basically, if I say that DNA is like this, okay, the DNA basically, when it is like this, and when it makes a new strand, when you have to duplicate the DNA, suppose a baby is being formed, you have to give the half of the DNA to them also, right? So how does the process work? So over here, your DNA is going to be segregated, separated, and a new strand is going to be formed. A new complementary strand is going to be formed. What can you see over here? One strand is old, so I'll say it is parental strand. One strand is new, so I'll say it's a new strand. So I get semi-conservative, only half of it is being conserved. This is known as semi-conservative mode of replication. And Watson and Crick basically spoke about this only. And my dear children, Michelson and Stahl came into the picture and they did the experiment on Escherichia coli, and the experiment goes on like this. Listen to me carefully. Over here you have the DNA. Now this Escherichia coli which is there, it was grown in a medium of N15 isotope. Okay, N15 isotope medium it was grown. And nitrogen is a component of your DNA, right? Nitrogen is basis, so it is a component of DNA. It takes up the nitrogen. So all the DNA which was being there, it was tagged with N15 isotope. Now basically when the E. coli, I centrifuged it in a medium of cesium chloride, CSCl, cesium chloride, it basically sedimented right over here. Why? Because both the strands were heavy strands. N15 isotope is a heavy isotope, so all of them get segregated out there. This is what I found. Now the next thing that I did is that same E. coli, my dear children, I made it grow in another medium containing N14 isotope. Now every 20 minutes it is going to do its division, every 20 minutes. So after 20 minutes when I saw that the new complement, so this strand is going to separate and you need to find the new strand, but new strand will not be having N15, rather it will be having N14 because you're growing in N14 medium, right? So the new strand came up over here. This time it is what? Green in color. This is for your differentiation, to your understanding. Okay, here also new strand is been coming up. So how do you find a new strand is coming up, and that is also N14? Basically again you did the centrifugation. When this time the centrifugation was done, It did not sediment over here, rather it sedimented over here. That means there was a pattern change. It's a hybrid thing that was forming. N14 is a lighter isotope, N15 is basically a heavier isotope, which is why you got a band over here. Now my dear children, for next 20 minutes, 20 plus 20, it's now 40 minutes. After that, when you saw, again this strand is going to differentiate. Similarly, this strand is also going to differentiate. The new strand which is there, it was obviously going to be N14 only, because you have not changed the medium. It is going to be remaining as N14. Now over here you do not have any you do not have the red thing, but here the parental strain is still there. Parental strain is still there, right? Every generation is still there. How do you confirm it? Basically, again, when you do centrifugation, you can find a new band forming over here for light strain, but still the hybrid strain is there. Why hybrid is there? Because the parental strand, whatever was there, it is being transferred and it keeps on transferring. And this shows semi-conservative mode of replication. This shows what? Semi-conservative mode of replication. This is super important. Okay, please remember it. E. coli divides after every 20 minutes, and there is a question on it. There is a question similar to it. There was one more experiment being done, and that was done by Taylor and his colleagues on faba beans. You also call this beans as Vishya faba. And over here, radioactive thymidine was used instead of what? The nitrogen isotopes. Radioactive thymidine was used by Taylor and his colleagues. Okay, and the work was done on faba beans. Please remember, this is an MCQ question that can be coming up. Now, the last thing, other important topics, as I told you, Hershey and Chase experiment, please go. Different types of RNA polymerase in eukaryotes. So, in the eukaryotes, my dear children, eukaryotes, they are generally very, very different rather than prokaryotes. Please know the differences of eukaryotes and prokaryotes. And over here, one such difference is eukaryotes have multiple RNA polymerase. There is RNA polymerase 1. The first priority is given to rRNA itself. Same name, no, that's why. rRNA. So, RNA polymerase 1 transcribes ribosomal RNA, or you also call this as rRNA. And there are different subunits of rRNA. Okay, different subunits of rRNA, and it goes like this: 28S, 18S, 5.8S. Okay, 28S, 18S and 5.8S. Now, RNA polymerase 2 is there. RNA polymerase, the second priority is basically to transcribe hnRNA, or what you call as mRNA, the major thing that you had to do, right? So, precursor mRNA is done by RNA polymerase 2. Now, RNA polymerase 3 is T, and it basically is there for transcribing tRNA. And apart from tRNA, you also have some 5S rRNA. Usually rRNA is done by that guy, but over here 5S rRNA is done by this guy, and small nuclear RNAs are also transcribed by this guy itself. So please remember, RNA polymerase 1 rRNA, RNA polymerase 2 hnRNA, RNA polymerase 3 tRNA. Apart from tRNA, 5S rRNA and also snRNA. Okay, and here everything ends with 8. 28S, 18S and 5.8S. Done. Madam, big fan from Kasaragod, Richie Richard Anthony. Hi dear. I didn't know I had fans in Kasaragod. So I see Malayalis gathering up over here. So good to see that. Okay, now one very important thing. Genetic content in different organisms, we studied it in the first slide itself. So that is also clear. Prokaryotes and eukaryotes, what are their differences in the terms of transcription, translation, everything? Just study it. Also, one more thing, cistron. Okay, so there are two types of cistrons. One is polycistronic organism and one is monocistronic organism. Polycistron means basically multiple genes will be there. This gene is going to code A, this gene is going to code B, this is going to code C. Multiple genes are going to be there in one particular unit. Okay, monocistronic means one unit will be coding for only one particular protein, not multiple proteins, only one particular protein. This is known as monocistronic. In the case of eukaryotes, we have monocistronic genes. One gene, one protein. But in the case of prokaryotes, so P for polycistronic, P for prokaryotes, they have multiple proteins being made. In one unit, multiple proteins are being coded up. Please remember, this question came in. For example, the question was, I don't remember. In pea plant, what type of thing that you see? Is it polycistronic or monocistronic? Remember, pea plant, it's a plant. Animals, plants, all of them are eukaryotes, and eukaryotes are monocistronic. are monocystronic. Prokaryotes are polycystronic. Prokaryotes means bacteria. Apart from that, there is nothing that you need to remember. Okay? Chandrakant, hello dear Chandrakant. Got it? Is this chapter clear? Is molecular basis of inheritance clear to everyone out there? If yes, can you give me a thumbs up? Everyone who is watching this video and who have understood this, can you just give me a quick thumbs up right now? Very, 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 very important. I am tired. Just a second, okay? I think I went somewhere. I'm not able to come back. Ah, yes, I'm back. Hi, ma'am. Okay, no thumbs up? No one understood what I was saying? Is that the case? Okay, then let's go ahead with the next set of things. So in this chapter, these are the important questions that you need to cover. If at all I have not covered it over here, please at least you go and cover. These are fixed questions that might come in your paper conceptually. Okay, it's not like question exactly will be coming up. Conceptually, these concepts are something that have been repeated so many times. So please go through all this concept. Remember, you will get the PDF of it. You just have to go to the description section and go check it out. Okay, go join the WhatsApp channel. It is definitely going to help you out. Works? Clear? Done? Okay, let's go ahead with the next chapter now, Principles of Inheritance and Variation. This chapter gives you a 2 marker and also a 5 marker, and if at all you want to grab them, listen to me carefully. Starting with the first question that we have, T.H. Morgan selected fruit fly for his genetic experiment. Why? Give 3 reasons. So what are 3 reasons? Why did T.H. Morgan select fruit fly? Why not anything else? Why not you? Why not me? So because fruit flies, my dear children, had certain very important features. First one was they could be grown in simple synthetic medium. They didn't say I want Parle-G, Boost, anything. They can be grown in simple synthetic medium. So it was very cost effective for them. Second thing, they complete their life cycle in 2 weeks. In just 2 weeks, if at all you see, they complete their life cycle. If you want to see any variation, you just have to wait for 2 weeks. That's all. Now, a single mating produces a large number of progenies. So many babies will be produced with a single mating itself. Now, you can do more experiments with all these progenies, right? So that is another thing. Now, there was a clear differentiation of sexes. The males and the females were easily distinguishable. The males and the females, you can find their differences very easily. This was also one more thing. So basically, in total, how many of things were there? These many things were there. Will you be able to remember it? I hope you do. Okay? Works. So T.H. Morgan selected fruit flies for his genetic experiment for this reason. Is it clear? Can I go ahead with the next question? Bacha Patti. Okay. The next question that I have over here is differentiate between linkage and recombination. What is linkage and what is recombination? Listen, linkage and recombination, both of the terms was given by T.H. Morgan itself. He did the chromosomal theory inheritance experiment. He wanted to prove chromosomal theory of inheritance, and basically when he did this experiment, he came to a conclusion for certain things. For example, when I talk about certain chromosomes, suppose the chromosome is like this. Okay, you have A gene, capital A gene over here, capital B gene over here, small a gene over here and small b gene over here, right? Now you have a different scenario where basically you have capital A over here, capital B over here, small a over here and small b over here. Now when you talk about linkage and recombination, things are going to be clear. Now in general, in sexually reproducing organism, you see a process that is known as crossing over. The chromosomes, the homologous chromosomes which are there, they cross over in between them. So suppose I exchange these parts. I exchange these parts. So what is going to be seen? Let me see that. Let me see that. So over here, I have capital, sorry, just a second, okay? Just a second, there is no space. So I'll make it like this and I'll show you. So when I did the exchange, small a came over here, capital A went over there, but small b is still over here and capital B is also over here. Now what I am finding is that I see a new combination. Small a was never with capital B and capital A was never with small b. This is what a new recombination, a new combination, and this is what I call as recombination. recombination. So basically, when the genes are very far from each other, the genes basically when they are very far from each other, they are going to show more new changes or more new combinations. So I'll say more recombinations will be there. And these type of genes are known as loosely linked genes. They are far away from each other. Now the next scenario is this, where basically they are very close to each other. Okay, very close to each other. Suppose this and this swaps up. What is going to happen? Basically everything looks the same. Small a, small b comes this side, and capital A, capital B goes that side. Do you feel there are newer combinations? No, there is very less combination. So I'll say that these are what? Tightly linked genes. They are tightly linked genes. And tightly linked genes are going to show lesser recombinations. Always remember that. The highest percentage of recombination which is there, it is what? 50%. It is going to be 50%. So when I say that two genes show 37.3% recombination, that means two genes are very far from each other, because 37.2 is very close to 50. When I say two genes show 1.3% recombination, I mean that two genes are very close to each other. That is why they are showing only 1.3. Okay, always remember that. Ma'am, is that PDF enough for tomorrow's exam? Yes. Yes, it would be more than enough. There is a PDF for all last minute revision I might have written, where everything is being covered, 1 marker, 2 marker for all the subjects. Please refer to that. Some MCQs, they were wrong. Just refer to the video also if at all you have any doubt in certain MCQs. Okay, it was marked out by my editor. So just check through that. Apart from that, it's all clear. Okay. Anmol Chopra, ma'am, do you know me? Sagar? Are you Sagar? Okay, so these are the things that you need to remember, and over here you are asked between linkage and recombination difference. So linkage is what? Tendency of genes located close together on the same chromosome to be inherited together. Recombination is what? It is a process by which new combinations of genes are going to be formed. Linkage, it will make sure that there is lesser variation. Recombination will make sure that there is newer variations out there or more variations out there. These are the things that you need to remember over here. Linkage and recombination, both of these terms were given by T.H. Morgan. And remember also one more thing, his student was there. This is an MCQ question. His student's name was Alfred Sturtevant. His student's name was Alfred Sturtevant. Now this Alfred Sturtevant came up with one very important term, and that is gene mapping. And that is gene mapping. What do you mean by that? The teacher gave linkage and recombination. So when the teacher said that percentage of recombination is this much, he got to understand that basically the genes are very far from each other. And with that understanding, he was able to locate the genes on the chromosome. More recombination, he was understanding that it was far away from each other. With that, he located the gene, and this is known as gene mapping process. Okay, I have tomorrow biology examination. Wishing you all the best, my dear. And as I said to everyone, please refer to the PDF which is there in the WhatsApp channel. If you have to scroll a bit, but you will find it. Refer to that. Watch the previous videos. It is definitely going to help you out. If at all you feel you do not want to watch this video as of now because you have exam tomorrow. Okay? Ma'am, can I go and read that PDF now? Yes, sure. Please. Please go and read the PDF now, and I'll recommend you watch the previous videos. If at all you feel, watch the video, read the PDF. Watch the video, read the PDF. It will be more easy for you. Okay? So you can put in 2x speed and watch. It is not going to, cause trouble. Yeah? Yes. Now, identify the symbols used in pedigree analysis. These are the symbols, my dear children, symbols which are there. So over here, I have two symbols that are usually being asked. That is, square is for male, circle is for female. This shows mating. Suppose there are two lines over here. This shows mating between relatives. This shows what? Mating between relatives. Okay. It's helpful for tomorrow. Yes, Sanju Shetty. It is. Okay, now over here when you see a male and a female is there, and it shows that the child is being diseased. How do you get to understand that? Because it is being colored. It is being colored. So please know all the symbols of pedigree analysis. There is one more symbol like this. This means sex is not specified. And suppose I am writing like this. This means 5 children are there, their sex is not being specified. If I color this, this means that this particular person has the disease. That's all. Okay? Got it? So these are the few things that you need to remember. Now, what is haplodiploid sex determination mechanism? Mention an animal which exhibits it. So please remember sex determination mechanism, all of it are equally important. I just have taken one question. So I can explain you two things. That is, there are two type of things. One is male heterogamety. Male heterogamety, and there is female heterogamety. Female heterogamety. What do you exactly mean by that? Heterogamete means different type of gametes. Heterogametic means different type of gametes. So listen to me very, very carefully. Listen to me very, very carefully, okay? So in the case of human beings, and also in the case of Drosophila, you have one very, very important thing, that is they show XX, XY type of sex determination, where the females are going to produce only one type of gametes, all of them carrying X chromosome. But the males, my dear children, produce two type of gametes. One is X and the other one is Y. Two different, that is why you call it heterogamety. What heterogamety? Male heterogamety. This is found in human beings and also Drosophila. There is one more example that you have, and that is of insects. For example, when you talk about grasshopper, they have XXXO type of sex determination. What does that mean? Here also females will produce only one type of gametes, all of them carrying X chromosome. But what about males? They carry either X chromosome or they carry no chromosome at all, no sex chromosome at all, and that is known as O. So two different type of gametes still being produced in the case of insects, for example grasshopper. So over here the example for male heterogamety is both of them. Female heterogamety is there. Female heterogamety means females are going to produce two type of gametes. Example, you just have one thing in your NCERT, and that is birds. And what is happening over there? ZZZW. Over here ZZ is for males. Males produce same type of gametes. One is Z, other one is also Z. What about ZW? This is females. Females produce two type of gametes. One is Z and other one is W. You can remember like this: W for women, and this is the only place where female heterogamety is seen, and that is in the case of birds. Now, as in the question that is being asked, you are being asked about what do you call this thing, that is, where is that? Haplodiploid sex determination. What do you mean by that? So it means that whenever an organism is haploid, that means it has only half the number of chromosome, it is going to be a male. If it is diploid, that means it has 2n number of chromosome, it is going to be a female. It is going to be what? A female. These points are super important. Okay. Now this is seen in the case of honeybees. So what is exactly happening over here? Honeybee has diploid content. The diploid content is 32 number of chromosome. Haploid, half of it will be 16. 32 will be there in female. If 16 is there, it is going to be a male. Now see, over here, female is 32 number of chromosome. When it produces gametes, it is going to undergo meiosis. Why? Because in gametes, only half number of chromosomes can go. So half of it is 16. Meiosis happened. In the case of male, it already had 16 only. Why will it undergo meiosis? So it undergo mitosis. It already is haploid, so it undergoes mitosis. It remains the same, 16 remains the same. Suppose this gamete and this gamete came together. 16 plus 16 is equals to what? 32. It is going to be a female. Suppose fertilization did not take place. If fertilization did not take place, it is going to be a male. It is going to be remaining the same. It is going to be a male. And this process is known as parthenogenesis. What do you call this? Parthenogenesis. It is known as partheno. It's not partheno, it's partheno, and R is also there over here. Okay, got it? In this particular case, you only tell me, does this guy have a father? No, Baba. It does not have a father, only a mother is there. But grandfather will be there. Why? Because mother was found from a father and the female gamete, from the father's gamete and the female gamete. So they have a grandfather but not a father. This is also there in your NCERT. This question, we never know when it will be asked. Okay, so please, haplodiploid sex determination, remember. Please remember heterogamety also. It is super important. Everyone who heard me now, okay, can you tell me female heterogamety is seen in which organism? Female heterogamety is seen in which organism? So I just discussed it right now. I want the answer, if at all you heard me. Ayyo. Can anyone tell me? Female heterogametry, no one can tell me? By the time you answer, I will also take some rest. Birds, yes, Rupashri is here. Oh my God, when I see my children, my regular children out there, I get so happy. And the new children who are there, I'm equally happy, and I'll be really happy if at all you come again and again, again and again. Hen, birds, hen, yes, 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 exactly. Now there is one more question, that is definition of point mutation, and give the example for point mutation. Point mutation means mutation is only happening at only one particular location, only at a single nucleotide. So listen to me very carefully. The classic example for this is sickle cell anemia, where you had just a second, where you initially had GAG. Now, at a single location, the nucleotide is being changed. Some mutation is happening. Now it is GUG. Ma'am, is that a problem? Yes, because GAG codes for glutamic acid. Glutamic acid, but this particular thing codes for valine. The amino acid itself will change. Do you think isn't a problem? Yes, definitely it is going to be a problem. So point mutation means a change happening at one particular point. Change in a single base pair of DNA is known as point mutation. Classic example for this is what? Sickle cell anemia, where glutamic acid is being was there, but now it is valine. And it is simple for you to remember. A comes before U, right? So A was there, it is replaced by U. Similarly, G comes before E, right? So G was there, it is replaced by V. Okay, done? Hi ma'am, how are you? I am super good, dear. How are you? How about you? You're good? Okay, so this is point mutation, and now we have the next thing. What are the conclusions drawn by T.H. Morgan crossing experiments in Drosophila with respect to linkage? I told you, if at all two genes are close together, they show lesser recombination. And I always tell you to remember in this way. If you and your friend are very close to each other, there is no chance another friend can come into your group. There is no chance a new combination can be formed. Similarly, if the genes are close together, there is no chance new combinations can be formed. On the other hand, if you and your friend are not very close, there can be a chance another friend can also get in. This is newer combination, right? So if you guys, if the genes are very far away from each other, they are not tightly linked, they are loosely linked, there are chances that new combination can happen. Always remember that. Okay, always remember, the higher the linkage, lesser the recombination. The lesser the linkage, higher the recombination. In other words, genes which are tightly linked will show lesser recombination. The genes which are loosely linked will show higher recombination. Okay, done. So when I say a particular thing is showing 37.3% recombination, that means they are far away from each other, because 37.3% recombination is too much recombination. 50% is the highest. So this means it is too much recombination. Got it? Remember that. Okay. Now, give reasons for the following. Let's go ahead. This is going to be the previous year model question paper. Give reasons for the following, and the first question goes like this. A simple cut results in non-stop bleeding in hemophilia affected individuals. Why do you think hemophilia affected individuals have non-stop bleeding? Baba, because you know, for clotting of blood, you need a very important protein. Suppose this protein is not being made. The gene for this protein is being mutated. The protein is not being made. Do you think clotting can happen? No. So the answer would be non-stop bleeding occurs when a single protein that is a part of cascade of proteins, a group of proteins. Okay, a single protein which is part of a group of proteins which is involved in clotting of the blood is what? Affected. This is what you see in hemophilia. Please write the name of the disease as well. It is there in the question itself, so don't worry. Now, next thing, Turner syndrome affected females are usually sterile. Turner syndrome affected females, why do you think they are sterile? Why do you think they cannot form babies? Because, my dear children, Turner syndrome means what? You do not have XX chromosome. This X chromosome is lost. There is not there. They have XO condition. That means they lack one X chromosome. If there are no two X chromosome, the female will be having rudimentary ovaries. The ovaries are not going to be fully developed. If ovaries are not developed, ovum will be formed? No, Baba. Ovum is also not going to be formed. If ovum is not there, do you think fertilization is going to happen? No, Baba. That is why we say that is going to be sterile. Females are usually sterile because they have only one X chromosome, leading to what? Underdeveloped ovaries and lack of normal gene production. Got it? That is the reason Turner syndrome patients or children, people who are there do not have fertility. Okay, and this is monosomy of sex chromosome. Monosomy, one chromosome is lost, loss of chromosome is known as monosomy. Now, in Morgan's dihybrid cross experiment in Drosophila showed that flies having genes for yellow body and white eyes exhibited less recombination. I always told you, when over here you are saying the genes for what? Yellow body. The genes for yellow body and white eye, they are showing less recombination. Means what? They are very close to each other. If they are very close to each other, wherever they go, they will go together only. Okay, they are going to go together only. Tightly linked genes is what you call them. I just explained you right now before some time. So they are tightly linked. So yellow body and white eye genes in Drosophila show lesser recombination because they are located close to each other on the same chromosome, enabling genetic linkage, or you please mention this, that they are what? Tightly linked. Okay, they are what? Tightly linked. This is super important information that you should not be missing. Next thing, inheritance of skin color in humans show different phenotypes. Phenotypes means physical nature. You and me, does your skin color match? No, Baba, we are all different. Why? Because in the case of human beings, the skin color is showing something which is known as polygeny. Okay, skin color inheritance is known as polygeny. That means multiple genes has an effect on our color. So I'll say usually Mendel said that one gene, one character. Mendel said one gene, one character, but here it is not happening like that. Here the thing is, suppose you have chromosomal combinations like this. Okay, and you have capital A, capital A, capital B, capital B, capital C over here. I say all of these genes basically code for my skin color. Multiple genes coding for one character. This is known as polygeny. Now listen to me. Here the genotype will be capital A, capital A, capital B, capital B, capital C, capital C. I say that basically if someone has a genotypic combination like this, they will be having the darkest skin tone. On the other hand, suppose I change the genes right up over here. I make it small a. Now the combination is going to be capital A, small a, capital B, capital B, capital C, capital C. Over here, this is going to be lighter. This shade is going to be lighter than the previous one. Okay, now over here, suppose it is small a, small a, small b, small b, small c, small c. This is going to have the lightest skin color. This is going to have the lightest skin color. Why? Because all the recessive characters are together. Okay, but here the thing that you can see is that basically multiple genes are affecting one particular character, and this is an example of polygeny. So inheritance of skin color is an example of polygenic inheritance. Okay, polygenic inheritance. It shows different phenotypes due to polygenic inheritance where multiple genes contribute to phenotype, leading to a range of skin color. That is why we have multiple skin color. Ayyo. At Karthik Kulkarni, make videos for KCET. Soon we are going to do that. Soon, really soon. Okay. Now, I hope that is clear. We have one more question. Accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why do you think accumulation happens in the case of phenylketonuria affected individuals, my dear children? Do you know that? Basically, in phenylketonuria, what happens is that one particular gene which is going to code for an enzyme, this particular gene is mutated. It is mutated, which is why this gene is not able to produce the enough protein that is needed. So basically, what is the name of the gene? What is the name of the enzyme? Phenylalanine hydroxylase. This is the enzyme which is there. Okay, it is responsible for the conversion of phenylalanine. This is an amino acid to something else which is known as, where did it go? To something else which is known as tyrosine. So this conversion was done by this guy, but now this guy is not there. Why? Because it got mutated, right? So what is going to happen? Phenylalanine is not going to be converted and it is going to be accumulated in the form of phenylpyruvic acid. You know what happens? When this accumulation happens, people get mental retardation. People will be having mental retardation because of the accumulation. What about tyrosine, my dear children? When tyrosine is there, tyrosine is not being produced, it is going to lead trouble in the formation of melanin, and melanin is the responsible for our hair color, skin color and everything, which is why since melanin is not going to be produced, we will also be having hypopigmentation. Hypo means less pigmentation. Hypopigmentation. You also say in your NCERT it is written reduced pigmentation in your skin and your hair. That does not mean reduced hair is there. Okay, hair will be there, reduced pigmentation is going to be seen. So basically over here, one gene is having multiple effects. Opposite of polygeny, this is known as pleiotropy. This is an example of pleiotropy. One gene, multiple effects. Okay, and here accumulation of phenylalanine is happening. Why? Because phenylalanine hydroxylase enzyme is not going to be produced, Baba. Okay, it is not going to be produced. So that is why there is a toxic buildup happening. Good evening, Shilpa, and hello, Chandu. Hello. Chandu ke cha chane. Okay, let's go ahead with the 5-marker question now. Few autosome-linked recessive gene blood disorders in human population. Among them, some are related to qualitative and some are related to quantitative problems of synthesizing blood proteins. What is this complex question? This question is complex, but it is actually simple. Listen to me very carefully. So, there are blood disorders, okay? In your NCERT, there are 3 blood disorders. One is hemophilia, okay? Hemophilia is X-linked disorder. It has some problems with the X chromosome. On the X chromosome, some gene is there that codes for protein and that gene is not produced. But the rest of the disorders which are there, one is sickle cell anemia and another one is thalassemia. Both of them are on your autosomes, not on your sex chromosomes, on the other chromosomes which you call as autosomes. So in the autosome, one gene is affected, which is why all this problem is happening. Okay. Ullas Yata, ma'am, you are too good. Thank you. Thank you so much. I am pretty sure that I could help you in some way, which is why probably you might be saying that. I'm so happy. That's a very cute gesture out there. Yes. So there is sickle cell anemia as well as thalassemia, which are other blood disorders and they're autosome linked. And the question is about them. Why is the question about them? I'm telling you that. So there is sickle. Okay, should I write like this? No. I'll just write sickle cell anemia and there is thalassemia. Okay, listen to me. We have chromosome number 11. On chromosome number 11, we have the genes for beta hemoglobin, beta chain of hemoglobin, or I'll say, yeah, I'll just write beta chain of hemoglobin. So listen to me carefully. Hemoglobin is made up of 2 things. One is your alpha chain and one is your beta chain. If any one of them is affected, obviously your hemoglobin is going to be affected. Hemoglobin is something that carries what? Oxygen out there. It has iron, it carries oxygen. And hemoglobin is found in RBC. Suppose I am saying the gene over here, it has some mutation. It has some mutation, and which is why if this is an mRNA and this mRNA is going to code for the protein, on the 6th codon of the mRNA, it was GAG first, but now over here in the 6th position, it is GUG. So this particular codon was coding for glutamic acid, which is an important amino acid. Okay, and here now it is coding for valine, which is another amino acid. Now if amino acid is changed, whole protein structure is going to be changed. If your whole protein structure is going to be changed, do you think beta globin chain is fine? No, Baba. Beta globin chain is not fine. It is not fine. The structure of it is having some problem. So I'll say the quality of it is reduced. I'll say the quality of it is reduced. Now if beta globin chain quality is reduced, it is going to form the same hemoglobin. The hemoglobin's quality is also going to be reduced. The RBC, which is going to carry this hemoglobin, its quality is also going to be reduced, and basically it will turn sickle in shape. It will be having the least oxygen carrying capacity. Oxygen carrying capacity is going to be the least, and not just that. Basically, because of its sickle shape, what is going to happen? It is going to stuck in between. This is sickle cell anemia. I hope it's clear. And always remember, A comes before U. So it was A first and it was replaced by U. Similarly, G comes before V. So it was G first and then it was V, and it is on the 6th position. This is a qualitative blood disorder. Now talking about thalassemia. Thalassemia is again it is on the chromosomes, autosomal chromosomes, and there it is going to be affected. So over here in thalassemia, you have to remember 2 things. Here we just studied about chromosome number 11 because your beta globin chain is on chromosome number 11. Here you have chromosome number 16 and also you have chromosome number 11. So your beta globin chains and your alpha globin chain. Alpha globin chains gene are located on 16th chromosome. Beta globin chains chromosomes are located on 11th chromosome. Your alpha globin chains have 4 genes. 1, 2, 3, 4. So basically these 2 are complementary and these 2 are complementary. 4 alleles in total. Here for them there is just 2. For beta there is just 2. Suppose I am saying this allele, okay, this allele is affected. That means the quantity of the alpha globin chain is going to be reduced. Okay, because there are other 3, they will be synthesizing, but quantity of it is going to be reduced. What about both of these genes going? Again, the quantity is going to be reduced because you have other two. What if all of them are not going to produce anything? There is no alpha globin chain going to be produced. Without alpha globin chain, you cannot make a hemoglobin as well. This is going to be a big trouble. So I'll say this is a major thalassemia patient. On the other hand, if at all you take over here, similarly in beta globin chain also, if suppose this gene is not working, this gene is mutated, still you will be having beta globin chain being produced. Why? Because you have another one which is active. Suppose both of them are gone, then beta globin chain is not going to be produced. Here, the quantity of it is being reduced, and that is why that's a big problem. So it's a quantitative trouble. Here it was a qualitative trouble, here it is a quantitative disorder. So both of them are blood disorders, but one is going to be qualitative and other is going to be quantitative. I hope you get it. So you have to write it down. So mutation in the HBB gene causes production of abnormal hemoglobin in this particular case, in sickle cell anemia. Defective hemoglobin leads to sickle cell red blood cells, causing a problem out there. Thalassemia, here it is caused by mutations in gene responsible for hemoglobin. It can be HbA1 or HbA2. So HbA1 has 2 alleles, HbA2 also has 2 alleles. These are 2 genes for alpha chain. And for beta chain also you have one particular thing, that is HBB. Any of this mutation causes quantitative troubles out there. Both of them are autosomal recessive disorder. In your NCERT, all the disorders are basically recessive disorders only. Only one example is given, and that is of myotonic dystrophy. That is autosomal dominant disorder. The pedigree chart is given of that, and that is the only one which is autosomal dominant. Got it? Any Bangalore students over here? Any Bangalore teachers over here? Me. Okay, and Shilpa, ma'am, please take some rest. I'll definitely take some rest after teaching you guys. Thank you for being so considerate. Ullas, ma'am, 0 to Hero series are enough for KCET 2026. Yes, but we are planning to cover the most important chapters only, but we are planning to bring something else also. Do not worry, okay? And it's free also. Don't worry, we are bringing it in. It's in process. And those of you who have not clicked on the like button, yes, click on it. And do not forget to share it with your friends. Okay, so yes. Now, going ahead with the next question, and that is, define aneuploidy. Give 2 examples of aneuploidy. Any idea? Aneuploidy, what is aneuploidy? Aneuploidy is basically when you have a loss or a gain of chromosome. Remember, you have 2 type of things. Okay. Mariam Salim, ma'am, is this enough for the next preparatory test? So I am talking about at least the major chapters. I am trying to cover it up over here itself. But if at all you feel you do not have much time to be in this live, I have a recommendation, and that is, please go and watch the previous live. Okay, there is a previous live. I am telling it because the people who are there, do not be worried. I am telling it again and again because there might be newer people coming and joining, right? So previously, I did a video. It is a recorded video. You might be able to see this, okay? Biology Preparatory 70 and 70 Part 2. This Part 2, my dear children, where did it go? Yeah, this Part 2 consists of chapters from microbes and human welfare to biodiversity and conservation. All 1 marker to 5 marker have been covered over there for these chapters, okay? Now, if you are looking for chapters from sexual reproduction in flowering plants to human health and diseases, there is another live that I have done before. For the first preparatory, the question remains, so that is the part, that is going to be the part 1. Where is it? Yeah. So first preparatory right there, and that is part 1. Over here you can see this. It has everything that you need, MCQs, everything out there. If you scroll down, yeah, that is going to be helpful for you. So you can watch that. You can get the PDF of it. You just have to join the WhatsApp channel. If you think that will be enough, you can go ahead that. Your explanation is so clear, ma'am. Thank you so much, dear. Thank you so much. It means a lot that good children are coming over here and telling about good things about me. I'm so happy. Okay. So aneuploidy is actually loss or gain of the chromosome, right? Yeah. So I was talking about Mendelian and chromosomal disorders. So yes. Listen to me. When we say Mendelian disorder, it means that one particular gene is affected, and that is why someone has the disease. When you say chromosomal disorder, it means that a chromosome is missing, or the structure of the chromosome is disoriented. That is what you call as chromosomal disorders. In your NCERT, there are just 3 chromosomal disorders which have been mentioned. One is this thing. What is that? Down syndrome. The second one is Klinefelter syndrome, and the third one is Turner syndrome. Apart from it, all of the others are Mendelian disorders. are what? Mendelian disorders. Ma'am, 3 marks, important question, please. So over here, I am dealing with this chapter. This chapter does not have 3 marks, only 2 and 5 marks. I'll come to the chapters which have 3 marks also, okay? Okay, so that is the thing. Okay, and aneuploidy is a reason why people have chromosomal disorders. Aneuploidy means basically loss or gain. Whenever you have a loss of chromosome, you call this condition as monosomy. Monosomy. Whenever you have a gain of chromosome, you call this condition as trisomy. So in this, you have 2 different type of diseases. Under monosomy comes one thing, and that is your Turner syndrome. Turner's syndrome. So in this particular case, the female should have been XS chromosome, but over here she does not have 1X chromosome. She does not have 1X chromosome. There is a loss of chromosome, which is why it will be XO. It will be just XO in her. So she will be having rudimentary ovaries. We discussed about it. She is not going to be fertile. She is going to be sterile in nature. Okay. Now, trisomy condition is where when you have an extra chromosome, and this can happen with the sex chromosomes, without sex chromosome. So listen to me very carefully. Suppose, okay, suppose over here you have a condition XXY. This X chromosome out there is an extra chromosome. So this is trisomy, example of trisomy. This is found in Klinefelter's syndrome. This is found in Klinefelter's syndrome. So usually we just have 2 sex chromosomes, but over here you have an extra X chromosome out there. This makes sure that this particular person who is having Klinefelter syndrome will be having masculine body. This is important. Symptom is important. Okay, they will be having overall masculine development, but there will be gynecomastia. That means formation of breast will be there. There will be development of breast over here in this particular condition. Okay, and they will be not showing spermatogenesis in their body also. Since there is extra X chromosome, there is no spermatogenesis that is going to happen over here. This guy is also going to be sterile. Okay, now you have one more condition, that is not the sex chromosome, that is the normal chromosome. What do you call the normal chromosomes? Autosomes. So in your body, we have 46 number of chromosomes in total. 44 of them are autosomes. They do not have anything to do with the sex of the human being. 45th one and 46th one are sex chromosome. It can be XX, it can be XY. Now over here, this one was basically sex chromosome related trouble out there. So I say that 44 autosomes will be there normally, but you will be having what? XXY condition. There will be XXY condition. Okay, this is karyotyping. This is known as karyotyping. Now apart from this, you can also write it like this: 47. Why 47? Because 44, 45, 46, 47. 47 chromosomes are there, and you can write XXY. That is also possible. If you write autosomes, it will be better. So 44 autosomes plus XXY, comma XXY is the karyotype for Klinefelter syndrome. Now when you talk about the next condition, I told you it's not just X chromosome, it happens in normal autosomes also, and that is in the case of Down syndrome. In the case of Down syndrome, the 21st chromosome, okay, the 21st chromosome, which is an autosome, it will have an extra copy. So instead of having just 2 of them, you have an extra copy. This is known as trisomy of 21st chromosome. And as I said, 21st chromosome comes under autosomes. So this time we have to change some numbers over here. So instead of 44 autosomes, this time it is going to be 45 autosomes, and it can be XX, or it can be 45A plus XY. It can be a male, it can be a female as well. This is the case of Down syndrome. Please know the symptoms of Down syndrome. There will be broad palm keys, there will be furrowed trunk, there will be mental retardation. All of this is super important. So I'll call this allosomal trisomy. I'll call this autosomal trisomy. Allosomal means allosomes are sex chromosomes. So allosomal trisomy, and this is also allosomal monosomy. Okay, allosomes are sex chromosomes. Ma'am, math preparatory 2 answer keys, please. Okay, do you want that? I'll definitely do that. Ma'am, please do bio first 4 practicals experiment and upload, ma'am. Apurva, we will be doing that really soon. Okay, so stay tuned for that. Really soon, we will be doing that in our channel. So if at all you have not subscribed to the channel yet, please, everyone who have not subscribed to the channel yet, subscribe to it right now, because it is going to be super helpful for you. Okay? Okay? Fine, I hope this is clear. So karyotyping should also be clear. How do you write the karyotype of monosomy? Turner syndrome, it is very easy. Listen, how many how many autosomes do we have? 44 autosomes, my dear children. Normal, right? Apart from that, you just have XO condition. So you can write like this. Or you can total it. How many chromosomes do you have? 44 and then 45. This is not a chromosome, right? So you write 45 and then you can write XO. So either way also it's fine. But I'll prefer this one. I'll prefer this one. I'll prefer this and this. Okay? Got it? Clear? So aneuploidy is the condition where organism has abnormal number of chromosomes, either due to gain or due to loss. Down syndrome and Turner syndrome has trisomy and monosomy, and symptoms of Klinefelter syndrome is all of this. Ma'am, there is book name SPR which is quite popular for both opinion on if you have a book to recommend. For 12th grade, for biology and all, I'll just recommend Griffith. That's more than enough. That's more than enough for what do you call biology. Okay. Hello, dear. So I hope this is clear. There is one more thing, that is polyploidy. Ouch. Just a second. Something fell. There is also something that is known as polyploidy. Polyploidy means the total number of chromosome, it becomes doubled. Okay. That is happening usually in the case of plants. It does not happen in the case of animals. Okay. Apurva, thank you so much. You're most welcome. Okay. And please share it with your friends also. Now we are going ahead with the next question, which is this, and that is represent schematically the result of one gene interaction between pure tall pea plant and pure dwarf pea plant. MQP3 question. This is important question. So one gene inheritance means you're talking about only one character. Monohybrid cross. Okay, only one character is being studied. And what is the character? That is height, dear children. So how do you do this? This is very easy experiment. We have done in class 10th also. Okay, so it goes like this. So it is like this. So parents, right? Parents over here. So I'm writing P. I will be writing P from now on to save some time. But you don't do that in your exam, okay? So capital T, small t, small t. Now gametes which will be there. Either half of it is going to come into the gamete, okay? So half chromosome, either it can be capital T, again it can be capital T only. Similarly, small t and small t. Now, the F1 generation, my dear children, when you cross this with this, you get capital T, small t. This with this, again capital T, small t. This with this, capital T, small t. This with this, capital T, small t. All of them, my dear children, are going to be tall. Why? Because there is capital T which is going to be dominant over them. So, all of them tall. What is a phenotype? Tall. Phenotype means physical nature. Okay, all of them tall. Now, Sangamesh, did you ask me something? Hello, dear Sangamesh. Okay. Yes. Okay. Charger. Okay. Just a second, children. I didn't charge, sorry. Who is there? No one is there. Okay, sure. Works. Okay. Fine. So I am here. Yeah. So now we are going to do selfing. Okay. We are going to do selfing. So what exactly happens in selfing, my dear children? What we need to do is have the parents, and we write selfing means crossing between both of them. So capital T, small t is there, capital T, small t is there. Gametes we have to form. So either it can be capital T, either it can be capital T, or it can be small t. Similarly, capital T or baba, it can be small t. Now you do the cross, and the crossing is going to be looking like this. So when you do the cross for F2 generation, this and this crosses, you get capital T, this and this crosses, you get capital T, small t, this and this, capital T, small t, this and this, small t, small t. Now you tell me the phenotypic ratio of this. This is important. Phenotypic ratio and genotypic ratio is important. My dear children, first we will find the genotypic ratio. Genotypic ratio means how do they look genetically. So 1 is capital T. So 1. 2 of them is capital T, small t. So 2. 1 of them is small t. So 1. 1 is to 2 is to 1 is the genotypic ratio. Done. Phenotypic ratio. How do they appear? Okay. How do they appear? So basically, 3 of them are going to be tall. Right? 3 of them are definitely going to be tall. 1 of them is going to be dwarf. So phenotypic ratio is going to be 3:1. Phenotypic ratio 3:1. Genotypic ratio 1:2:1. Got it? So please write this and come, and you will get your marks. Ma'am, Karnataka board has released a handbook. Can I study that to score good grades? Mairaj, I just have to check it up, what handbook you're talking about. When I check it up and I see the things, I can tell it to you. Okay. Modin Sahib, hello dear. Ma'am, Aadhaar card video ke reply Madi, please. Rushit, dear, I'll reply to you, okay? I'm just waiting for a clarity on things. Remember, just think, if I give you a wrong answer, it will be a very, very big problem. As far as I know, your name, your DOB, everything should be matched with the with the mark sheet. I know your concern is the parent's name. I saw that particular thing, but I'm unsure about it. The parent's name, if it is changing, it's a problem or not. Either one thing that can be done, since you have time, please mail to KEA. There is an option to mail to KEA, right, or to ask KEA board. Please ask them itself. That will be the first thing. And also from my end, I'll be definitely doing it. I have been stuck with a lot of things right as of now, but I'll be soon coming with a lot of things. Okay, we'll help you with that. Okay. Fine. Let's go ahead with the next question. And that is this. Categorize following as either autosomal or sex-linked disorders: hemophilia, thalassemia, phenylketonuria, myotonic dystrophy. Categorize following as autosomal or sex-linked. Hemophilia is sex-linked. Thalassemia, phenylketonuria is autosomal. What about myotonic dystrophy? Ma'am, this is not there in our book. What are you saying? It's there in your book. You might have seen the pedigree chart, and that pedigree chart, it is given for myotonic dystrophy only. So basically, 1, 2, 3 are autosome. This is autosomal dominant disorders. So D for dystrophy, D for dominant, you can remember like that. Okay, this is sex-linked and recessive, and this is autosomal recessive. Okay, now what conclusion you arrive after careful observation of the following given diagram? So this is the picture given to you in your model question paper, and you are asked, what do you observe from this? So basically, GUG valin, I already told about sickle cell anemia, right? So sickle cell anemia, sickle cell anemia, which is there, it is caused because of the change in one particular base pair. Okay, one particular base pair, GAG was converted to GUG. So you just have to talk about point mutation, and you say just about sickle cell anemia. That will be more than enough. Now there's a question: identify the disorder by the following symptoms. Individuals with overall muscular development but with gynecomastia, that means development of breast. I already told you overall muscular development, but you have development of breast, then it has to be, then it will be what? It is going to be gynecomastia. It is going to be Klinefelter syndrome. It is going to be Klinefelter's syndrome. Got it? This is your MQB question. And with what I realize is that your MQB questions are not going to be, repeated as such. Rather, the concepts related to the questions which have come might be coming in. Okay, so that is the thing that you need to be sure of. So all your concepts around this should be clear. So here I have the answer. I will be giving you the whole PDF for all the chapters. Just join the WhatsApp channel. Do not be worried. You will get it. Okay? Okay. Now, explain thalassemia as an example of Mendelian disorder. I already told you. Thalassemia, why is it a Mendelian disorder? Mendelian disorder means a gene, when its problem is happening, it is going to cause trouble, right? So that is Mendelian disorder. So thalassemia also, when a gene is not there, basically it is not going to code for either alpha gene or beta gene, and that is why the whole problem is happening. So that is why I call this as a Mendelian disorder. And how do I explain it? Thalassemia is an autosome-linked recessive blood disorder transmitted from parents to the offspring when both parents are heterozygous carriers. Okay, both of them should be heterozygous carriers. Now this is important. There can be questions on the basis of it. So I won't be explaining all of it because I already explained it, but I really want to help you, show you something. Okay, and that goes like this. So listen to me very carefully. If at all I am saying that in sickle cell anemia or phenylketonuria or thalassemia, there are two parents who are carriers. Will the child have the disease? The question is that, will the child have the disease? Let's see. So suppose HBA is a normal gene. HBS is the mutated gene. Similarly, HBA is a normal gene. HBS is the mutated gene. When this is there, it is dominant over this, and which is why the person will be just a carrier, will not have the disease. This is also the same thing. This person is also the carrier. Now, these two carriers, when they come together, do you think they can? Okay, they will be having a disease. Let's see that. Suppose this and this gene came together. HBA, HBA. The person is going to be super normal because both the genes are fine. HBA, HBS. This person will be again a carrier because he have a normal gene over here, there, and the mutated gene over there. So the normal gene will overshadow the effect. Suppose this and this came together. HBA and HBS. HBA and HBS, again carrier. What about this and this? HBS, HBS. this person is going to be diseased. So out of 4 people, 1 person is diseased. That means 1 by 4th. That means 25% chances are there for a person to be diseased. You get it? So this is why we say that before producing babies, the people should get their genetic checkup being done. Why? Because you might see it normal, but what happens? You might be a carrier for any disease, and there are 25% of chance for a male or a female to get this disease also in the case of autosome-linked recessive disorders. Got it? Ma'am, how to join WhatsApp channel? So there is a link given in the description section of this particular video. Okay, there will be a link given over here. You just have to join that. That is to get lecture notes, PDF of this lecture. Join Deeksha KCET WhatsApp channel. You just have to join the channel. Okay, works? Okay, fine. So let's go ahead with the next question. The next question is schematically represent incomplete dominance by taking inheritance of flower color in Snapdragon as an example. So how do you take that? Snapdragon and incomplete dominance, what do you mean by that? Until now, Mendel said that capital T is dominant over small t. What if I say that sometimes this is not true? Not every time capital T is going to be dominant over small t. There are going to be some changes out there. So how do I show it? I am going to show it with the help of incomplete dominance, and it goes like this. So just a second, okay? Okay. Capital R means red color. Small r means white color. Okay, and capital R shows that it is dominant trait, right? Suppose I am crossing capital R, small r, small r. What is the thing that I am going to get? I will be getting all of them are going to be capital R, small r, whatever I do. Right, now what do I expect? Capital R is dominant over small r, so I expect all of them to be red. But is that seen? That is not seen. That is not seen. They are not going to be red. Instead, they are going to be pink in color, which means that this particular gene is not completely dominant over the other gene. It shows incomplete dominance. And things did not stop there. They did selfing also, to just test it up. Okay, they did selfing. Now when they did selfing, let's see what happened. So over here you have to make like this, okay, and explain. Please make sure you write the parents. Where is it? Just a second, something has happened. Okay, no, come on, PC, work. PC is not working. Just give me a second. Just like your teacher, your PC is also tired. Come on. Okay. What happens if I close this up? I do not close it up. What happens if I try it now? Guys, just give me some seconds, okay? I really want to fix this up. So for fixing it, I need to come. Yes. Yeah. So we were over here. Ayyo. Okay. So now it won't go. Okay, fine. So now we are going to do selfing. So selfing, let's see how it goes. Okay. So it goes like this. Capital R, small r, capital R, small r. Okay. Now here you put the gametes. When you cross this with this, Baba, you get capital R, capital R. When you cross this with this, you get capital R, small r. When you cross this with this, again capital R, small r. And when you cross this with this, you get small r, small r. Right? Now over here, one is capital R, capital R. It will be red in color. But what about them? They are capital R, small r. So it is incompletely dominant. Still they are going to be pink in color. What about this? There is no dominant letters out there, so it is going to be white in color. So if at all you talk about phenotypic ratio, one is red, two of them are going to be pink, and one is going to be white. Phenotypic ratio is going to be 1:2:1. Until now we saw it was 3:1, the phenotypic ratio. Three of them were tall, one of them was dwarf. But that is not happening over here. What is the genotypic ratio? Genotypic ratio, one is capital R, two of them are capital R, small r, and one of which is the plant that you are studying? You are studying snapdragon, or basically you are studying Antirrhinum majus. The name of the plant is snapdragon, or you also call this Antirrhinum majus. It is also known as 4 o'clock plant. Okay, works? Yes, Ashwini. Yes, Kavanna. Super good. Awesome. Okay, ma'am, please take rest. Abhi, thank you so much, dear Kishan. Ma'am, you guys don't teach in a right way, guys. You will be very quick while explaining. Dear, you can stop me right there whenever you feel that you are not understanding. I am explaining faster because I feel that you do not have much time. Okay, that's the only reason. Okay, so Classy, hi dear. Hi, hello. Okay, let's go ahead with the next question that we are having, and this is again an MQP question. This was there this year, so you should know the answer for this. Okay, let's go ahead. It goes like this: sex-linked recessive. You have to identify the genetic disorder. So sex-linked recessive disorder occurring in about 8% of males and only 0.4% of females, leading to defect in the cone of the eye. What is this disorder? What is this disorder? Okay, so 8% of males are affected, but only 0.4% females are affected. This disorders which affects the cone of your eye, it is none other than color blindness. Color blindness. Over here, males are the one which is mostly affected here. Why males are affected? Why this cola very D? So listen to me carefully. It's an X-linked recessive disorder, and the same goes with hemophilia also. Okay, why males are mostly affected over here is that basically X-linked recessive disorder means only when 2X chromosome having the effect comes together, then this particular person will be having the disease. Now in the case of males, you do not have 2X chromosome, you just have 1X chromosome. So if 1X chromosome is affected, then this guy will be having the disease. What if the female is like this? The female will still be safe because you have a normal X chromosome. This will overshadow the effect. But in the case of males, it is not like that, which is why males are more prone to have this color blindness. More prone to have this color blindness. Okay. Now listen to me very carefully. I am going to give you some instances that is going to happen. I am saying that the male is normal. There is a normal male and there is a carrier female. Carrier female means she has the gene but she is not diseased. She does not have the disease because the X is there to overshadow the effect. Now suppose this and this came together, you got XX, the female daughter is normal, this and this came together, the female daughter is now a carrier, this and this came together, it is XY, the male is also pretty normal, this and this came together, it is XCY, here the male has the disease. So out of two sons out there, one son is definitely going to have the disease. 50% chance is there for a son to get the disease, whereas the female, they do not have the disease. She can be a carrier, she can be normal, but she does not have the disease. When will a female have the disease? A female will have the disease when the father is diseased. When the father is diseased, the female will also be diseased, and the mother should at least be a carrier. Should at least be a carrier. So in that case, let's make the female diseased over here. So here, the male is having the disease. The female is at least a carrier. In this particular condition, there are chances that this X chromosome and this X chromosome, when it comes together, the female will be getting the disease. The female might be getting the disease. There are chances over here the female might just be a carrier also. But if at all you want to make the female disease, at least this condition should be there. Okay, suppose the male is diseased. Only the male is diseased. Ayo, just a second. Okay, only the male is diseased over here. The female is normal. Do you think the female will have the disease? No, Baba. The female will not have the disease. Why? She can be just a carrier. Whatever you do, it will be XXC, XXC. She will be just a carrier. Here the males are also fine. But if the mothers are at least a carrier, the male will definitely have the chance to get the disease. If mother is a carrier, okay, suppose here the mother is a carrier. there is a chance for a male to get the disease because the mother can contribute this chromosome, then it will be XY. If the mother contributes this chromosome, then it will be XY. So either way, if at all you see, the males are more prone to the sex-linked recessive disorders out there. Okay. Hi Aman. Got it? So please remember that. Now in the case of sickle cell anemia, we say, sorry, not sickle cell anemia, hemophilia, we say that females basically do not get hemophilia itself. Okay, why females do not get hemophilia, my dear children? The reason being that, suppose hemophilia is a disorder where basically your blood clotting is affected. If your blood is not clotting, do you think you will live? No. Color blindness is something that you can live with. You cannot see the colors, green and red, but at least you can live with that disease, right? But in the case of hemophilia, it's not like that. So in the case of hemophilia, suppose I take the female is a carrier. Okay. The male is not having the disease. The male is not having the disease. In this particular condition, XX, it will be normal. XX, she is going to be a carrier. XY, he is going to be a normal person. This and this, he is going to have the disease. Now, if I want to make a female have the disease, I need to have the male to have the disease first, because there is a need of X chromosome out there, right? Now, over here, you only think one very important thing. Will the male be alive until the age of reproduction to basically reproduce with this particular disease? No, Baba. The male would not be alive for that. Which is why we say that the chances for the females to get the disease is very less. Even if I make a carrier for female, I cannot consider this combination to happen. Female carrier can live, but male who is having one chromosome affected cannot live until reproducible age, which is why they cannot reproduce a child who is going to have this. So always remember, in hemophilia and sickle cell anemia, there's a slight difference. Sorry, hemophilia and color blindness, there's a slight difference in this. Males are mostly affected in both of them, but in the case of hemophilia, females, there are lesser chances for them to be born because of this. Okay. Hi Tarak. Hi. Hello. So yes, now sex-linked recessive blood disorder affecting a single protein involved in clotting of blood, we know it is hemophilia. That is the only disorder where you have this. Now, inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine. It is phenylketonuria. It's an example of pleiotropy as well. I discussed about that. Next thing, autosome-linked recessive blood disease, substitution of amino acid in beta globin chain of hemoglobin. Substitution is happening in beta globin chain, and that is going to be sickle cell anemia. That is the only where point mutation is being seen, right? Next thing, autosome-linked blood disease, reduced rate of synthesis of globin chains of hemoglobin. Reduced rate, quantity is reduced. It is going to be thalassemia. Quantity has the term T, and T is for thalassemia. You can remember like that also. Okay, Kavana, yes dear. So these are the diseases which are there. This is from your NQP2, so you can have an understanding of it. Okay, works? Okay. Now, if that is clear, can everyone give me a quick thumbs up? If this particular question is clear, can you all give me a quick thumbs up? And everyone who is watching this video, if at all you have not hit on the subscribe button yet, click on it, and my dear children, click on the like button, so that when you click on the like button, this video basically gets propagated to all of them who are preparing. Okay, ma'am, can I refer only question bank released by Karnataka boards to score more marks, ma'am? Please reply. Yes, question bank is really good source. You can go ahead and solve questions. If at all you solve 100% of questions, obviously it makes sure that your concept is super clear. So if you have time, you can do so. But do you think you have time for completing the whole thing? If you have time, please go ahead with it, okay? Let's go. Now, represent. Thank you so much for giving me the thumbs up. Lots of love to all of you guys. Okay. Represent schematically the results of two-gene interaction by taking seed shape and seed color in pea plant. Two things. So this is going to be a dihybrid cross. When do we call it dihybrid? When basically okay, I'll explain it to you with the help of this, and with this explanation you are never going to forget anything about dihybrid cross. So let's get ahead with this. So basically, my dear children, I am going to take two things. One is shape and the other one is color. shape and color. Shape is round and wrinkled and color is yellow and green, right? So I am taking suppose pure round and yellow plants and pure wrinkled and green plants. Okay, pure round and yellow, pure wrinkled and green plants. Fine, they are not dihybrids. They have two characters. I am studying two characters, but they are not hybrids now. You will call them hybrids when all the alleles are going to be this different. I am going to explain you that. So listen to me. So how do you make the gametes for this? This is a question that usually children have, and this is a place where you guys do ratification, where you do by hearting. This time onwards, never do by hearting. So listen to me carefully. The scenario is like this: one capital R over here, one capital R over here, one capital Y over here, one capital Y over here. Similarly, one small r over here, one small r over here, one small y over here, small y over here. Now you need to make the crossing. So from this gamete, from this chromosome, only half is going to come, either this one or this one, but anyway it is going to be capital R only. Similarly from here, this pair also, only one is going to come. Whatever you do, it is going to be capital Y only. So gametes are going to be many, 4 of them actually, but it is going to be capital R, capital Y. Similarly from here also you get small r from this and small y from this. Do you have any other combination that can happen? No, Baba. There is no other combination that can happen. So these are the things that you get. Now when these two gametes come together, what is a new thing that you will be finding out? Capital R, small r, capital Y, small y. This is going to be the new thing that you are going to find out, and it looks like this. So one chromosome capital R, another chromosome small r, this is you got from the other parent. One chromosome capital Y, another chromosome small y, this you got from the other parent. Now it looks like this. This is your first generation. All of them are going to show the dominant character, that is what? Round and yellow. Round and yellow. But are they like this? No. This is the first dihybrid that you have got. Capital R, small r, both of them vary. Similarly, capital Y, small y, both of them alleles are varying. So this is what you call as dihybrid. Now we are going to do the selfing. Let's do the selfing and let's get the answers. So when you do the selfing, it looks like this: capital R, small r, capital Y, small y, capital R, small r, capital Y, small y. So when you do selfing, it is like this: capital R, small r, capital Y, small y. Let's make the gametes. My dear children, from this pair, capital R can come. From this pair, capital Y can come. This is a gamete. From this pair, capital R can come. From here, small y can come. This is another gamete. From this pair, small r can come. From here, capital Y come. This is another gamete. From here, small r can come. From here, small y can come. This is another gamete. So how many different type of gametes do you have? 4 of them. Similarly, you will get 4 of them from that side also. Now if you align them accordingly on both these sides, what do you get? 4 here, 4 here, and you cross. Capital R, capital R, capital R, capital Y, capital R, capital Y. You make the cross, you get 16 combination. How many of them? 16 of them. And if at all you study all these 16 of them, you are going to see a ratio, and the ratio is very simple. It goes like this: 9:3:3:1. 9 of them are going to show the dominant character, which is what? Round and yellow. Which is going to be round and yellow. 1 of them is going to show the recessive character, which is going to be wrinkled and green. Wrinkled and green. Now you make the new combinations. This 3 and 3 is the new combination out there. And what is it? Let's see. So round was with yellow, but this time round is going to be with green. Here wrinkled was with green. Let's make wrinkled with yellow here. Wrinkled is going to be with yellow. This is the combination that you are going to get. This is the phenotypic ratio. Now if at all you are asked, how many different type of phenotypes do you get in a dihybrid cross? 4 of them. These are the 4 phenotypes. How many unique genotypes do you get? Unique genotypes means without any repetition. So many things are repeated over here. 9 genotypes are there. 9 unique genotypes are there. Always remember. You can remember like this: 9, 9. Both of them starts with 9. Okay, phenotypic ratio is this. 4 phenotypes are there. Genotypes are 9 of them, and you might get this question also: how many non-parental variety come? 3 plus 3, 6 of them are non-parental variety. Now if at all suppose a question is there where you are being asked that there are 64 plants, and out of them I want to find out how many of them were non-parental. How will I find it out? So basically there is a formula which you can use. How many of them are there in total? 16 of them are there in total. Out of 16, how many of them are non-parental? 3 plus 3, 6. 6 of them are going to be non-parental. 6 by 16 into the total number of organisms which are given. If at all you do the calculation, you will get your answer to be what? 24 of them are going to be non-parental out of 64. This is a question that may come. Please know about it. Okay, it's an MCQ question which has come. So I just explained that to you right now. Okay, got it? So please remember all this thing. If at all it is clear, yes, genotypic ratio 1:2:1. Exactly, super, 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 super. Awesome, awesome. Awesome, Kavana. Philomena Pereira. Give me a kiss. Okay, guys, is it clear? Is everything clear? Can you give me a thumbs up if it is clear? If it is clear, can you give me a thumbs up? I'll go ahead with the next thing. This is supposed to be the last question. The next question is going to be the last question of this particular chapter, which is what do you call principles of inheritance and variation. Ma'am, please make it simple. Numan Inamdar, dear, I cannot make it more simpler than this. How are you? I'm really good. I'm really good. Thanks for asking. How are you guys? So children, I'm assuming that it is clear to you guys, and with that we go to the last question of this particular chapter, and that is Mendelian disorders may be autosomal or sex-linked, dominant or recessive, homozygous, heterozygous. Pedigree symbols are used to analyze these disorders in human population. Mention the genotypes of the following pedigree symbols. So, autosomal dominant. My dear children, autosomal dominant, and over here a square is there which is colored up. That means this man has the disease and it is an autosomal dominant disease. So if I am assuming this, so here I have a dominant allele over there, that means it can overshadow the effect of recessive allele, and if at all this dominant allele is problematic, it can cause the trouble. So this can be one genotype. The next genotype which can be there, it can be this. Both the alleles are dominant. Both the alleles are dominant. In this particular condition, this particular man can have the disease. Now there is autosomal recessive, and over here it is mentioned that this female is not diseased. The disorder is autosomal recessive. That means if at all the person has to be diseased, both the alleles should be recessive in nature. Both of them are autosomal. But over here we do not want the person to be diseased. That means we have to do something ulta of it. How do? We just write this. So AA, dominant, whenever dominant allele is there, it is going to be normal. Whenever it is going to be like this person will be having the disease. Since this person does not have the disease, you have to write like this. Okay, works? Okay, now going ahead, just give me a second. Okay, Soumya is here. Hello ma'am, because of you I got 64 marks in first preparatory. Oh my god, thank you so much. Thank you so much for coming over here and telling that up also. It means a lot. Awesome. And Numan, please make it simple. The pedigree one, I mess with that every time. I'll try to make it as simple as possible, okay? Okay, come listen to me. So, you have to look at this also, and you have to see this particular thing also. So, the next one is sex-linked dominant. Sex-linked dominant means basically over here, I'll say, this particular person does not have the disease. This particular person does not have the disease. But if this person need to have the disease with this, it should have been XAY, or it could have been XA. That's all. This is a male, right? So this is a male, so X and Y should be there, and the person should have had one dominant allele, that is capital A out there. But over here this person does not have the disease. That means we have to make the opposite of it. And what will be the opposite of it, my dear children? The ulta one. X small a y. Small a means this particular person is having the recessive allele. You just have to mention that. Now next thing, sex-linked recessive disorder. Sex-linked recessive disorder means if at all a person is having the recessive allele, they will be having the disease. Or the female has both the alleles as recessive, she will also be having the disease. So over here that is being asked, the male is affected, the female is also affected. That means the condition is going to be this for the male, and this is going to be for the female. Okay, this is going to be for the female. Ma'am, please reply my question. Kavan, I didn't see you the question. Please, can you ask me once more? So sorry. I'm so sorry. Ma'am, MCQ question for question bank is enough. Yes, listen. I had solved so many question bank questions also in the Brahmastra series, and with experience I'm telling you, MCQ questions, you can expect some of them to be coming up, but most of them could be new as well. But at least the thing is that the questions, more questions you practice, at least you are very much thorough with the concepts, and that will be very much enough for you to score good. Okay? Works? Is that clear? Fine. Pedigree 1, Numan, is it clear to you? You just have to look this, and then you will be able to. I know it's a bit tricky. Take time to solve pedigree related questions. And also in pedigree related questions, if at all you see that parents are not affected but the next generation is, skipping of generation is being seen, it means it is recessive disorder. If in every generation one person is affected, okay, in every generation, if one person is affected, for example, I say like this. This means every generation, if one person is affected, I'll say it's dominant disorder. It's going to be a dominant disorder. Okay? Madam, pass Agudvanan. Nitin, you will pass. Okay? Kavana, ma'am, I dream to take 70 in biology. You will get 70 in biology, dear. You'll get 100 in biology after your practicals also. So guys, every one of you who is watching this video, who is basically biology student, we are bringing in practical videos for you really soon. So subscribe to the channel, because I know your exams are also going to start up. So please stay subscribed, okay? And these are the other questions. If at all I have missed out anything, please be sure and solve all these questions. Super important. Okay, go through all these questions if I have missed out anything. Okay? Fine? Cool. Guys, then should we go ahead with human health and diseases? Do you need a break? Can I take a 5-minute break? If it's okay, can I take a 5-minute break? Will you be doing biotechnology? Soumya, no, I have recorded it. It's in the recorded video. Can you please watch that? I think that will be better for biotechnology. Listen, here also I am going up really speeded up, right? So I would not be covering that up. Please watch that particular video. It is definitely going to help you. Trust me, I'm telling you. Okay? Okay. So children, I'll just take a 5-minute break and I'll come. My back is paining too much. Okay, so just give me 5-minute breaks. 5-minute Kit Kat break. For all of you, go eat some good food and come for 5 minutes. Just have some snacks. I'll be back at 7:20. Okay? I'll be right there. Be back. Bye. so the Kit Kat break is officially over. So let's get ahead with the video once again. Ready? Ready, steady, go. So this time we are going to be a bit more energetic. So why is it that in my screen I'm not coming? Yes, I'm there. Okay. So, hi dear, hi Numan. Okay. So let's go ahead with the chapter that is Human Health and Diseases, and the question goes like this. First one, mention the causative organisms for the following diseases. The first thing that you have is Ascariasis. Ascariasis is caused by Ascaris lumbricoides, my dear children. It's a round worm. Typhoid is caused by Salmonella typhi. It's a bacteria. Filariasis is caused by filarial worm, that is known as Wuchereria bancrofti or Wuchereria malaria. And my dear children, it is spread by the mosquito bite. Remember that also. Okay, so it is a nematode disease, but it is spread by mosquito bite. Okay, and ringworm, it seems like a worm is causing the disease, but no. But it is no. It is caused because of fungus. So fungus is the problematic person over here. So you have trichophyton, epidermophyton, both of them are fighting, and you have microsporum, small spores. You can remember like that. Okay. Now malaria is there. Malaria is caused by Plasmodium vivax or Plasmodium falciparum or Plasmodium malariae. Three of the things are there. The most malignant form of malaria, dangerous form of malaria, is caused by P. falciparum. Okay, it is going to be caused by P. falciparum. So please remember. So the causative organism for all these diseases are this, and please know the symptoms of this as well. Ma'am, I got 63 in preparatory because of this channel. Oh, that's so sweet. Thank you for coming and telling that up. Ma'am, application-based questions is pretty tough, ma'am. Please help. So application-based question becomes easy when your concepts are really strong. So we have to make the concept strong. If concept is strong, it will be easy. Otherwise, it is going to be difficult. Well, I have solved a lot of questions in the previous thing. So if you join the WhatsApp channel, you'll get the PDF. Okay, just refer to the PDF. Just see the questions, if at all you're able to do or not. If not, then please watch the video. Okay, videos also previously have done it. Arun, when do you take PU1 class? Very soon. Okay, very soon we will be doing PU1 classes as well. So this is the first question. This came in your this thing also. What do you call that as? Model question paper also. Now, the next question is, name the group of genes that have been identified in normal cells that could lead to cancer. My dear children, it is there in the normal cells, but it can lead to cancer. Do you know the name of this genes out there? It is known as proto-oncogenes. What do you call them? Proto-oncogenes. So over here, listen to me carefully. So normal gene is there. You have some proto-oncogenes, normal genes, which are doing some normal function out there. They're chilling around. But one day, some mutations happened over there. And when mutations happen over here, basically, they turn into cancerous genes. And they cause what? Cancer. So name the group of genes that have been identified in normal cells that could lead to cancer is basically none other than proto-oncogenes. This is important. Now, which technique was useful in detecting of cancers of internal organs? 3-marker questions. So you have to talk about it. So first thing is biopsy and histopathology. First thing, suppose there is some growth anywhere. So what do? We take some cells out of it. This is known as biopsy. We cut that region and we take some portion out of it. Now we do studies on it. Okay, we study it under the microscope. This is known as histopathology. So biopsy and histopathology is the first thing. Now next thing, CT scan. For CT scan, my dear children, it is known as computerized tomography, and here you are going to use X-rays, and these X-rays are not pretty good. They are kind of dangerous only. So these X-rays are going to give you the 3-dimensional image of your internal organs. If you're specifically asked about internal organs, what is the technique? MCQ question, it will be CT scan. Okay, now MRI is there, magnetic resonance imaging, and I'll say that it is pretty much safer. Why? Because here you are not using X-rays. No ionizing radiations are used. Non-ionizing radiations as well as magnetic field is going to be used, and it is for detecting any problem in the living tissue. So that is MRI. Apart from that, you also have X-ray, normal X-ray, that is known as radiography. So these are the few things that you need to have for detecting cancer of internal organs. Man, will there be hot questions from this chapter? Hots. Not from specifically from this chapter, from principles and molecular basis of intelligence you might be having. Okay, so CT and MRI. Yes, exactly. Now, why are cancer patients often given alpha interferons as a part of treatment? Why do you think? Because you know, in cancer patients, they do chemotherapy, they do radiotherapy, lot of things. The normal cells are dying out there along with the cancer cells also. So normal cells mean immune cells are also dying. So who will provide them immunity? There should be someone to modify the immunity, right? And that is the role of alpha interferons. Alpha interferons are given to cancer patients because they are biological response modifiers. That means they increase our functioning of the immune system. So always remember that it enhances the immune system. Okay, so these are the few questions from your MQP3, question number 39. Yes. Now next question, name of the drug, its source and nature is given in the table below. Find A, B, C, D. So A, B, C is only there. Ma'am, really confusing. Numan, what is confusing there? Tell me. What is confusing? So if you sit without any expectation, if you just listen to me, all your confusions are going to go away. Don't be sad. When you guys are sad, I feel sad. Okay? Okay. So over here, if you see, a source plant and then nature is given. So the name of the drug you have to tell, the source plant is already given. Papaver somniferum. From Papaver somniferum, what do you get, Baba? You get two drugs. One is heroin and the other one is morphine. Heroin, which is there, it is a depressant. It depresses down, it slows down your body's function. So your answer for this question is going to be heroin. Now next, name of the drug is cannabinoids. Okay, cannabinoids, a group of drugs. What is the source plant? It is going to be cannabis sativa. Cannabinoids, my dear children, they are going to affect your cardiovascular system. Your cardiovascular system is going to be affected by cannabinoids. That is the second group of drugs. Now you have cocaine. That is a third group of drug coming under coca alkaloids. The source plant is Erythroxylum coca. Now they are asking you what is the nature. So usually they basically what do you call? They give a sense of euphoria. You feel so happy. Apart from that, why you are so happy? Because it messes up with your dopamine transport. Every time when you scroll your mobile phone, know what is going to happen? Basically, you are getting dopamine. So over here, the same thing is happening. Your dopamine transmission happens regularly. There is no stoppage, which is why we feel a sense of immense happiness. You call this as euphoria. That does not mean that you need to take it, because, my dear children, it also causes hallucination. You will see cows started flying. You also start flying along with the cow. So hallucination can also happen because of cocaine. Okay, so it is basically where is the answer? Answer is not that. Yes, heroin is the answer for the first one. Cannabis sativa is next one. And effects on the central nervous system. Yeah, that also you can say. It directly affects on the central nervous system in this particular case. Okay. Ek minute. Can you explain all the drugs? It will be easy to revise. Yes, I can do that. And I think I have brought that list. Just a second, okay? I think I had made a list. Yes, you guys are lucky. Do you know that? Because the previous time I didn't bring this particular PDF. Yes. So listen to me carefully. If at all you listen to me carefully for the next 5 minutes, probably every question related to drugs will be answered very easily within seconds. Ready? Ready? 1, 2, 3, let's go. Opioids are the first drugs, okay, first group of drugs. Over here you have two things. One is morphine and the other one is heroin. How do you get morphine? You get morphine from the latex of the poppy plant. Poppy plant is known as Papaver somniferum. You can remember Tupperware bottle, okay. So Papaver somniferum, this is the plant, this is the pod. Here you get latex, milk-like thing. From the latex, what do you get, Baba? You get one very important thing that is known as morphine. You know, morphine is given by the doctors to the patients who have underwent surgery, because morphine is a painkiller and it's also sedative. People used to sleep after getting morphine. That does not mean if at all you're not getting sleep, you can take it up. No, that's not a thing. It's only given by the doctors. Increased dosage of it can lead to death also. So morphine is there. We got from Papaver somniferum. That is there. Now, if at all this morphine is made to undergo acetylation, it is going to convert into something that is known as heroin. Heroin has different names. It is known as smack. It is known as heroin itself. It is also known as diacetyl morphine. Diacetyl morphine. Everywhere, three names are there. in the case of heroin. Now heroin, both of them are going to basically slow down a little bit things. Heroin especially, it is a depressant, it slows down your body as well. Now you might ask, how do you take heroin? Well, you should not take it, but I'll tell you. Mode of intake is either snorting or basically injection. Snorting or injection is the only way you can take it up. Okay, and it is white, it is odorless, does not have any smell, it is bitter, crystalline compound out there. These are the basic things that you need to know. Now, where does it affect? This group of drugs, opioid, where does it affect? Two places. Your central nervous system, CNS, that is your brain and your spinal cord. Also your gastrointestinal tract. So CNS and gastrointestinal tract is something that you need to remember. Now I'll revise, and this revision should be quick. Opioid is there. You get it from papaver somniferum. There are two drugs, morphine and heroin. Morphine is already there. It is a sedative and a painkiller given by doctors to the patients. If at all this particular morphine undergoes acetylation, you get heroin. Heroin is a depressant. It slows down your body function. Heroin is basically taken up by snorting or by injection. It is white, odorless, crystalline, bitter compound substance out there. It is also known as SMAC. It is also known as diacetyl morphine. Now, where do these drugs affect? It is going to affect your central nervous system or your gastrointestinal tract, and it is going to get down. Okay, that's all. This is about opioid drug, and remember the structure of morphine. It looks like opioid is basically from O. So here also it is O. It looks like O. Okay, ma'am, please slow down. This is really hard to digest. Okay. Okay. Is it clear? Only when you say it is clear, I will be going ahead with the next one. If it's clear, give me a thumbs up. I'll be going with the next one. Okay? The effect, listen, in the group of drugs, there are two groups of drugs which affects your central nervous system. One is opioid and the other one is coca alkaloids. That is basically cocaine and things like that. So here, along with CNS, it is also your gastrointestinal tract which is being affected. Children, if it is clear, can you give me a thumbs up? If it is not clear, can you ask me to explain again? I can explain. It's not a problem for me. Okay. Numan says it's clear. Okay, thank you. Hello ma'am, say it is confirmed, it will come or not. It might definitely, there are chances it might come. From drugs, at least one time it come. Nagaraj is here, hello. Karnataka Seal Farm, thank you so much for giving me that thumbs up. Means a lot, guys. Now the next group of drug after opioids is this one, cannabinoids. Okay, cannabinoids. Cannabinoids you get from cannabis sativa. Where do you get it from? Cannabis sativa. From the inflorescence. Every part of this plant can give a drug. Whether it's leaf, whether it's root, whether it's stem, whether it's pod, anything, it can give you a drug. Now, there are multiple drugs under cannabinoids. Okay, marijuana, the most famous drugs that you might heard of, they all come under this. Okay, marijuana, hashish, ganja, charas, bhang, all of it, they come from this. They won't ask you from which part. All the parts are basically used here. Now, how do you intake cannabinoid? There is inhalation. Either you inhale it, or basically you might have seen people of Punjab drinking bagno. So there is oral ingestion. Basically, they mix and eat it up. That is also possible. Okay, that is also possible. Now, cannabinoids, where does it affect? This is a drug which is unique. It does not affect your CNS. It is going to affect your cardiovascular system. That means basically your blood, your heart, cardiovascular system is going to be affected over here. And cannabis sativa looks like this. It looks like this, like the fingers out there. And the picture which is given, as I told you, morphine's picture is there and this picture is there. In this picture, you can think that there will be some stairs also. Some line of stairs is also there in the case of cannabinoids. So I'll just do one more time. Okay, I'll just do one more time revision. Listen to me carefully. Cannabinoids you get from cannabis sativa. It looks like this. Over here you have stairs. Okay, and basically it does not affect your CNS but affects your cardiovascular system. How it is taken? Inhalation or basically oral ingestion. You mix it and drink it. Okay, hashish, marijuana, ganja, charas, everything you get from different parts of this particular plant. And basically also remember one very important thing, and that is sports people. Sports people mostly take up this particular drug, which is why in your NCERT it is clearly written cannabinoids are abused by sports people or sports person. Please remember this. Provide PDF. PDF will be definitely provided. Ashwin Wagmare, dear, I did a previous video on fixed questions. You can go and just write fixed questions biology. In the front page itself, you will be able to find it. Okay, there are certain questions, go through each and every question chapter wise. Now listen, the concepts are repeated, not the question exactly. The concepts are repeated, so you have to be strong with the concept. Okay, works? Clear? Okay. Ma'am, do these diagrams come for the exam to draw? No, Baba. You just have to identify it. That's all. You just have to identify it. Works? Cool? Okay. Next thing. Now we have coca alkaloids. What do we have? We have coca alkaloids. And what effect does it do? It affects your central nervous system. What effect does it do? It affects your central nervous system. So basically it is going to interfere between the dopamine transmission out there. Okay. So I'll come back again. Coca alkaloids, you get it from Erythroxylum coca. And remember, this is native to South America. You need to remember the place, South America, not North America. This question has come. Erythroxylum coca from South America. Now, common name is coke or crack. They ask you, heroin is known as smack, coke or crack. Heroin is smack, this is coke or crack, CC only. Now, it is usually being snorted, and the effect it has, its effect is on central nervous system again. Okay, but here specifically between dopamine transmission. Between what? Dopamine transmission. So it is going to do two things. One is it is going to act like stimulant. That means it increases euphoria, it increases energy, happiness, everything. High doses of it can also lead to hallucination. You will start seeing cows flying. That is what I was talking about. So coca alkaloids, you get it from Erythroxylum coca, and the common name is coke or crack, and the mode of intake is snorting. Okay, it is a stimulant, it increases euphoria or energy, and high doses of it can also lead to what? Hallucination. It affects the central nervous system, especially the dopamine transmission. Got it? Is it clear? Hello, Ibrahim. Hello. Okay, so listen, one more very important thing, and that is other plants causing hallucinations. So there are three plants in total. One is Erythroxylum coca, one is Atropa belladonna, and the other one is Datura. Atropa belladonna, Datura, and also, as I said, the other one. So over here, Atropa belladonna looks like this. Okay, sorry, Datura, it looks like this. So there is a cup-like structure, funnel-like structure over here. It is known as Datura. Okay. Okay, got it? Clear with all the drugs out there? Now there are few more drugs, the other drugs which are there, not categorized under anything. One is this: barbiturates, amphetamines, and benzodiazepines. Barbiturates, amphetamines, and benzodiazepines. It is used for depression as well as insomnia, and it is often being abused as well. So barbiturates, amphetamine, benzodiazepines are basically used to treat depression as well as insomnia. If at all someone is not able to sleep, they are given these particular drugs. BAB. Okay, BAB. You can remember like that. Okay. Now, effects on tobacco. So there are few things, few other drugs, as I said. One is nicotine. If in your question it comes that some drug is responsible for increasing the blood pressure, just understand that name, that drug's name will be nicotine. It is not cannabinoid, even though cannabinoid affects your cardiovascular system. It is going to be nicotine that affects your blood pressure, because nicotine, my dear children, it stimulates the adrenal glands, and adrenal glands is going to release adrenaline and noradrenaline. Okay, and it leads to increased heart rate and also blood pressure. Also remember this very, very important thing. Got it? Ma'am, what do you mean by hallucination? Hallucination basically means you are starting to see things which are not actually there. Okay, not actually there. That is what you mean by hallucination. Call? Works? Now, I think that's done. Those are the drugs that you need to remember. Apart from that, in your NCERT, there is anabolic steroids given at the end. Okay, anabolic steroids, just know anabolic steroids have different problems in the case of males as well as females. In the case of females, it causes In the case of females, it causes this thing. What do you call this as? Mood swings will be there. Okay. There will be acne. There will be acne. All these things are, and basically the level of hormones is going to be imbalanced out there. In the case of males, again, the testicle size is going to be reduced, mood swings is going to be there, acne is also going to be there. These are the few things that you need to remember. At the end of the chapter it is given, please make sure you go through it. Okay. Ibrahim, just a second, okay. So basically, dear, I had made a question, just a second. If at all you see the video, and if you go down a bit, where is it? If you move down, ayyo, just a second. Why does this happen? Videos. Just give me a quick second, okay? I think there's a glitch happening out there. I am ready. There is a fixed question that I did. You can go and see those questions, okay, if at all you want. Where is it? Yeah, there is a fixed question video that I have done. If at all you want, you can go and watch this video. Please make sure you solve all the questions, and please make sure you do it nicely, as in the concept should be super clear. Because the concepts are going to be repeated. It's not the question which is going to be repeated. That has never happened. So concepts are going to be repeated. Make sure you go through these questions again. Super clear? I hope it's clear. And over here itself, my dear child, I had done, just give me a quick little second. Where is it? Okay. So I had, okay, that's gone. I had done a video on fixed questions also, part 2. You can watch that video also. Okay. Hiyo, Damodin, dear, I have already covered that particular chapter. Please, can you go and check it out? It's in the initial part of this particular video. Okay. Ma'am, you're so pretty. Thank you so much, dear. And you are also pretty, dear. And how, ma'am, can you explain BT cotton related questions? So BT cotton in general can be asked, as in how does it work and things like that. If you wait for some time, I'll just complete this chapter and I can explain you about BT cotton. Okay? Works? So this is the thing about drugs, my dear children. Now we go on to the questions that we were solving, because there are another important questions that you should be knowing, and that is this. So describe any 3 properties of cancerous cell. What is the 3 properties of cancerous cell, dear? 3 properties of cancer cell is, first one, uncontrolled growth. It goes on growing. Second thing, loss of contact inhibition. So basically when cells divide, they cannot continuously keep on dividing. Okay, generally when normal cells divide, suppose when these cells divide, when they come in contact, they stop the division. They stop the division right out there. But over here in the case of cancerous cell, that is not going to happen. They are going to show something that is loss of contact inhibition. Loss of contact inhibition basically means that it is going to keep on dividing, even though if it comes in contact, it is going to keep on dividing. That is the next thing. Third one is metastasis. What do you mean by metastasis? Metastasis basically means that at one particular place, suppose the cancer cells are there, they are moving into different places of our body and dividing up there, causing trouble up there. This is known as metastasis. Okay, they can spread to distant body parts and keep on dividing up there. Got it? Got it? Works? So these are the 3 properties of cancerous cell. Now, what are lymphoid organs? Write the functions of the following in response to immune system of our body. This is also an important question. Lymphoid organs which are there, they are of 2 types. They are of 2 types. One is your primary lymphoid organs, and the second one is your secondary lymphoid organs. The primary lymphoid organs are your bone marrow and your thymus. Your bone marrow and your thymus. Your secondary lymphoid organ is going to be what? Spleen, lymph nodes, spleen, lymph nodes. Apart from that, what do you have? You have tonsils, you have pairs patch. Now, why do you call it secondary and primary, ma'am? That can be a question. So listen to me very carefully. Okay. So over here, in this particular case, primary lymphoid organs are the place where the lymphocytes, that is the B and the T lymphocytes, are going to be produced. They are produced there. They are going to mature there. Okay, and basically they become antigen sensitive. Please remember these things. They are going to be produced there, they mature there, and they become antigen sensitive. That means they know that whom do we have to fight with. They know their enemies. But do they start fighting? No, Baba. They do not start fighting. They just know it. Where do they fight? They fight over here when they reach to secondary lymphoid organs. So here they become effector cells. Here they become effector cells. So this can be a question. Lymphocytes, where do they become effector cells? A, B, C, D options would be given, and you should be able to tell the options which one is the correct answer. Okay, so bone marrow is the place where your B and your T lymphocytes are being formed. B lymphocyte stays over here itself for maturation. What about T lymphocytes? It goes to thymus for maturation. Thymus is found over here behind our ribs. Okay, behind the sternum, not the ribs, behind the sternum in the central position. And when we age the thymus, the size of it gets reduced. The size of it is going to get reduced. Okay, that is with thymus. Now, talking about the secondary lymphoid organs, we have spleen. Spleen is just near our stomach. And spleen is known as graveyard of RBC. All the dead old RBC comes and dies over there. But basically, spleen also have high amount of lymphocytes. And these lymphocytes are waiting for any pathogen to come so that they can kill them up. So spleen is a secondary lymphoid organ because their fighting is happening. Now, over here, in the case of lymph nodes are found everywhere in our body. When you go to doctor touches over here. Why? Because lymph nodes are over here. Lymph nodes are having so many lymphocytes. So if at all any infection is happening, they fight and there will be an inflammation. That is why doctor touches over here and checks. Okay. Tonsil, which is there, they also have high amount of lymphocyte which is there for fighting. Peyer's patch is found in our small intestine. They also have high amount of lymphocytes where they fight. Now you have MALT, mucosa associated lymphoid tissue, given in your NCRT. It said that they occupy 50% of the lymphoid tissue. In MALT is mucosa associated lymphoid tissue. Mucosa is what? A layer of our intestinal tract, a layer of our tract. From here to here, this mucosa layer is there. So anything that is related to the mucosa layer, for example, tonsils, for example, Peyer's patch. Okay, all of them will be having, all of them comes under MALT, and basically they are responsible for fighting against diseases. Okay, got it? Fine. Jyoti is asking graft. Dear, graft means basically anything that you take from outside. For example, my kidneys are not working fine. I took yours. Okay, and basically my body rejects it. Why? Because it rejects it because it's foreign to me. And their T lymphocytes is something that acts very fast. Okay. Got it? So you have to mention about bone marrow as well as the thymus properly. I've written everything over here. When you get the PDF, you will be able to see everything as well. So please write it and please bring it. Please write it and take it in your examination. These are the lymph nodes, as you can see over here. It's everywhere in your body. And this is your spleen, dear. Okay, this is your spleen just towards your, this is your spleen just towards your stomach. Cool? Fine. Graft rejection, okay, got it, fine. Now, the most important and the most famous question that usually comes up from this particular chapter is replication of retrovirus. That means replication of HIV virus. And how do you explain this particular question? It's pretty much simple. Listen to me carefully. The virus or the HIV virus is, HIV virus which is there, it is basically known as human immunodeficiency virus. Okay, it is known as human immunodeficiency virus. It has RNA in its body. Okay, in its cell it has RNA. Now basically we have DNA, right? We have DNA. This particular virus goes and affects our cell. Our cell has what? DNA. If it wants to use our cell's machinery, it has to form DNA. But Baba, it had RNA. How will it make DNA? It has machinery for that. It has an enzyme that it brings with itself, and that is known as reverse transcriptase. What do you call it? Reverse transcriptase. With the help of reverse transcriptase, it is going to reverse the process of transcription. Dear children, you always have studied DNA forms RNA. The reverse of it is known as reverse transcription, and it is done by reverse transcriptase. So RNA, which is introduced by the virus, will now turn into a DNA. Now this DNA is going to be integrated into our genome with the help of another enzyme that is known as integrase. You do not need to learn it if at all you feel it's difficult for you. Okay. So over here, I have taken up over here. I have taken it over here. The viral DNA is now inside. The viral DNA gives information to form the viral RNA, the viral protein, everything. Now all of it is going to assemble and it is going to go to other cells and affect them. Okay, RNA to DNA by reverse transcriptase. Exactly, Jyoti. Okay, so this is how it usually goes in. So always remember, it has RNA content, and reverse transcriptase is a very important enzyme out there. And also remember, the first cell that it mostly affects is the macrophages. It is the macrophages. This is known as the HIV factory. Why? Because HIV viruses usually comes and affects them first and increase their number. But their main target is not macrophages. If you are thinking it's macrophages, it's wrong. It's not macrophages. It is going to be, the main target is going to be TH lymphocytes. What is TH lymphocytes? It is basically T helper lymphocytes. T helper lymphocytes are something that are going to help other cells and tell that, hey, you have to be immunologically active. Hey, you have to be active. So if this guy is not there, who is a mediator, my dear children, your immune system is going to go weak. Your immune system is going to go weak. Get it? So helper T cells are super important. So give the schematic representation of replication of retrovirus. It goes like this, and there is a question in your MQP that you need to solve right now. It looks like this. Okay, so over here they have given you retrovirus. Virus infects the normal cell. Viral RNA is introduced into the host cell. Now reverse transcription happens. After that, what happens? After reverse transcription happens, viral DNA is going to be formed. Yes, now we know the answer. Now the viral genetic material in the nucleus of the host cell. Now what is going to happen? Viral DNA incorporates into the host genome. Then virus will produce its own proteins, own materials out there. That is what you have to write. Viral protein, viral RNA is going to be produced over here. Then virus comes out of the host cell and new viruses can infect. So the question goes like this: fill in the missing data in the boxes labeled as 1 and 2. Now you know the missing data. Label 1 is viral DNA is being formed over here. After reverse transcription, viral DNA is being formed. The next question is what? Viral components are being synthesized. So basically over here, once the viral DNA incorporates into the host genome, viral components are going to be formed right over here. Okay, got it? So this is something that happens. Now give an example for retrovirus. Baba, it is HIV, human immunodeficiency virus. Name the disease caused by this retrovirus. AIDS, acquired immunodeficiency syndrome. You are acquiring it throughout your lifetime. It's making you immunodeficient, and basically it's a group of symptoms, which is why you call this as syndrome. Now name the diagnostic test used for this particular disease. The diagnostic test is what? ELISA. ELISA stands for enzyme linked immunosorbent assay. What do you call it? Enzyme linked immunosorbent assay. This is the diagnostic test as well. Got it? Got it. BT cotton, I'll explain it. Don't worry. I have not forgotten it. Okay? The question is not over. There are two more questions. Okay. Differentiate between innate and acquired immunity. What is the difference between innate and acquired immunity, Baba? Every one of you, come on. What is the difference between innate and acquired? Innate, for example, the skin. Is there anyone in this human race who thinks that? Incorporate means join, right, ma'am? Yes, exactly. It joins up. Okay. Innate, is there anyone in this world who is born without skin? No, we are all born with skin. It is there with us from our birth. Skin is there. We started crying and we came out. Tears are there from our birth. So basically these are innate immunity, something that is there with us from our birth. And it is non-specific. It is not specific that I will only act on this particular thing. No, it's common for all. It's acting on everything out there. Acquired immunity is basically something that you acquire during your lifetime. For example, any pathogen came into your body, an infection happened, you reacted to it, your body produced some antibodies, all of it is acquired immunity. You are going to acquire it throughout your lifetime. Okay, so there are some differences present from birth. Here acquired is developing after infection or some vaccination. It is non-specific. Here it is specific to particular antigens. There is no memory cells being produced, but in acquired immunity, my dear children, there are two cells which are being involved. Okay, there are two cells which are being, yes, HCl is also one such thing that comes under innate. Now, a homework for you guys that you have to do from your end is please go look for all the barrier methods under innate immunity. There is physical, that is your tear, skin and mucus and everything. Physiological, where you have your tears, you have your HCl being produced, and there is cytokine barriers, where basically, cellular barriers, where you have some cells such as PMNL, polymorphonuclear leukocytes. Okay, they are there from the birth. Okay, and such as macrophages, they are also there from the birth. And you have cytokine barriers, that is interferons. Now, interferons is a very important question. Virus infected cells releases what? So, virus infected cells releases interferons. This is different from alpha interferons that you give in cancer. So, this interferons goes and tells the other cells that, hey, the virus infection has happened over here, you guys have to be safe. So, they all come under barriers of innate immunity. Go through it. Examples are super important. Now, acquired immunity, as I was saying, they are specific to particular antigens. Why they are specific? Because over here, two type of things are happening. One is humoral immunity, and the other one is cell-mediated immunity. Humoral immunity and cell-mediated immunity. In humoral immunity, my dear children, you have one very important thing to remember here. Antibodies are involved. Antibodies are going to mix up with the blood, and that is why you call this humoral. Humor, blood is basically what? Humor. Okay, blood is basically a humor. Okay, humor means not that humor, humor, it's just humor as in fluid. Now, antibodies are mixed, but who is producing it? It is the B cells. So, B cells, whenever it encounters an antigen, a problematic person, it is going to produce two things. One is antibodies against it. Second, it is going to produce some memory cells, so that it has a memory that this thing has happened. Now, the next time it happens, I have to be more careful. That is what you mean by this. Now, the next thing, that is B cell, B lymphocytes, which maintains humoral immunity. You have cell-mediated immunity. That is done by T lymphocytes. Over here, why cell-mediated? Because these T lymphocytes go and tell the other cell that you have to be active, and this is how they build the immunity. And this T lymphocytes, my dear children, whenever they encounter some antigen, they either go eat it up together, you call this as cytotoxic T cells, or they help the immune system to be active, that is T helper cells. And basically, they are mostly the ones which are important for graft rejection. Okay, they are the ones involved in graft rejection. As I was saying, Jyoti asked me a question that if at all, I take a kidney from someone else, my body rejects it. Why? Because my body feels it is foreign, it is an antigen, it's problematic. That is why. Okay, so acquired immunity have these two things, and it is very specific to particular antigens, and it has immunological memory. Here, innate immunity examples are skin and phagocytes. Here in acquired immunity, it is antibodies and T cells as well. Got it? Fine. Now, there's a question. Refer to the below given diagram and answer the questions. Name the molecules trapped in the lymph nodes responsible for the activation of lymphocytes that bind to A and B site. So they are saying some things are trapped in lymph nodes. What are those things? Antigens. And they are going to be trapped. They are going to basically combine at the A and the B site. Why A and the B site? Because these are the antigen binding site of the antibody. The antibody is made up of two things. Two heavy chains that you can see over here and two light chains. And this particular portion that you see is known as the antigen binding site, where the antigen is going to come and bind. Okay, and that's very specific to the antibodies. Please make sure you go to the picture. So what does H2L2 stand for in antibody? Heavy chains 2 and light chains 2. And remember, how many antibodies do you have, dear children? You have 5 antibodies. Gamed. Okay, it is gamed. The first antibody that passes from the placenta of the mother to the baby will be IgG. Second one, which passes from the milk of the mother to the baby, it will be IgA. So A for Amma and A you can remember like that. The antibody which is released during allergy, okay, the antibody which is released during allergy is E, allergy, antibody E. And always remember, in allergy you have certain cells which are very active that are known as mast cells. So mast cells, on its surface you have certain antibodies, and these antibodies are nothing but IgE. Whenever something comes and attaches over it, that is antigens, the mast cell is going to release two things. One is histamine and the second one is serotonin. Both of them are going to make sure that the blood vessel is dilated. If the blood vessel is now like this, all the other cells will come. Oh my God, what happened? What happened? It causes inflammation. That is why in allergy you have inflammation. Ma'am, then how do we treat allergy? There are three things that you need to keep in your mind. Against histamine, you need to give antihistamines. Antihistamines. Okay. Now, antihistamines, and apart from that, you also give adrenaline. And then you also give steroids. Please remember, this is how you're going to treat allergy. This is how you treat allergy. Okay, is it clear? Is it clear to you guys? Fine. This is super important. Please remember everything. And yes. Now there's a question over here which I missed. Mention the cells involved in accomplishing acquired immunity, B lymphocytes and T lymphocytes. I discussed about that. Now, yeah, name the cells involved in humoral and cell-mediated immunity. This is this year's question, MQP1's question. Humoral immune response, it is B lymphocytes, as I told you. Cell-mediated, it is T lymphocytes. There is one more question, and that is regarding active and passive immunity. Active immunity, my dear children, is when, okay, listen to me carefully, this thing also. Then we will talk about beta carotene. Active and passive. Active and passive immunity. Active is basically when your body is actively producing, okay, the antibodies and things like that. It is actively involved in the process of immunity. So suppose you are getting an infection. When you get an infection, your body reacts to it by producing what? By producing some antibodies, right? Now it can be slow response, but it is there. Now, if not infection, if not natural infection, suppose you are getting vaccination. In vaccination, what are you given? You are given weakened pathogens, some pathogens that cannot cause any trouble in you. You're just given some portion of it. Why? So that your body knows that something like this can be coming up and you need to produce antibodies. So when you're given vaccination, your body produces antibodies. Right? This response is slow, but next time the same pathogen will be coming up, it will be faster. But anyway, your body itself is producing antibodies. Are we giving antibodies? No. So this is active immunity. It can be ignited with the help of natural infection or vaccination. Now there is passive immunity. Your body is not actively producing anything over here. You are getting antibodies. For example, there is natural passive immunity, for example, from the mother to the baby. Through the colostrum, the first milk, the mother is passing IgA antibody, ready-made antibodies. Is the baby producing it? No, it is being passed. And now placenta to the baby, again the mother is giving IgG, right? So this is natural way. Now suppose I am saying that a person have got snake bite. Okay, a person have got snake bite. In snake bite, do you wait? When you go to the doctor, do you wait for that particular person to produce antibodies in themselves? No, Baba, that person will die. Doctor will give ready-made antibodies. Here again, you are not active. You are given ready-made antibodies. This is also an example of passive immunity or passive immunization. Second thing, tetanus injection when you are given antitoxin. This antitoxin is nothing but antibodies, ready-made antibodies. This is example of passive immunization. So whenever your body is not active, it's giving, it is passive immunization. Whenever basically your body is producing its own antibodies, it is known as active immunization. Always remember that. And hepatitis B vaccine was first made in East. This is also given in your NCERT in this chapter. Do not forget it. Okay. Okay. Now comes the thing that you were asking me for, which was, first of all, these are the other important questions. If at all you want to check, please check and please solve these questions. malarial parasite life cycle is also important. Okay, please check that question as well. I had discussed that in in the other thing. But if you need help with malarial parasite, I can explain to you once again. And now, Bt toxin. How many of them are there for Bt cotton? How many of them are waiting for Bt cotton for me to explain Bt cotton? Please tell me. I can explain it to you. Antivenom. Okay, then I'll explain. I think someone was waiting for Bt cotton anyway. So listen, I am not teaching biotechnology chapter in this particular live, but I can explain since that was a doubt. So Bt toxin, if at all we see, so there is a bacteria Bt, Bacillus, my hair. Okay, there is a bacteria that is Bacillus thuringiensis. Okay, Bacillus thuringiensis. Now this particular bacteria produces inactive toxin in its body. Now suppose one day a little caterpillar came and ate a leaf which had this bacteria on the top. Now this bacteria is inside what? Inside the gut. Inside the gut of the caterpillar. Now inside the gut, specifically if I say mid-gut of the insect, what is going to happen? This particular bacteria which is there, it is going to get favorable condition. By favorable condition I mean alkaline pH. A basic pH is going to be there, and in this alkaline pH what is going to happen? The inactive toxin which is there, it is going to become active in nature, and when it becomes active in nature, it is going to cleave open, cause perforation over here, and is going to make this caterpillar die, this particular organism die. Got it? Got it? This is how it works. Now Bt toxin is very much specific. First of all, Bt toxin is being produced because there is a gene in this organism, and this gene is known as cry gene. And there are two types of cry gene that you need to remember, which might come in your examination, and it goes like this. One is cry 1AB. Sorry. 1. Okay. Cry 1AC, and the other one that you have is cry 2AB. Now, listen to me carefully. First word is C. First word is C. So I write cotton. Cotton's first word is C. Second word is from B. So I write cotton bollworm. Okay, now there is one more cry gene, and that is known as, cry 1AB. So cry 2AB was done, cry 1AB is over here. So there is only one, and it also starts with C and it ends with B, and that is corn borers. Ma'am, both of them are starting with C and B. How do we remember it? Listen, cotton bollworm is a bigger name, which is why it needs 2 genes. Corn borers is a small name, it is why it needs 1 name only. It needs 1 only. So 1 is enough, and the first and the last letter is being mentioned over here. So you should not mess up with C over here, which is why I have mentioned B. So cry 1AB for corn borers, cry 1AC and cry 2AB for cotton bollworms. If at all you think that this is helping you, you can pick it up, or if you have any other formula to remember it, you can. But this is generally how I remember it. Cry 1 and 2. 1 comes before 2, so 1 and 2. And here also is just 1. Okay? Works? Clear? Is it clear to my chutku people out there? If clear, can you give me a thumbs up, everyone? Everyone, if it is clear, just give me a thumbs up. Model question papers 32 and 33. If at all you tell me the question, I probably might be able to do. Okay, fast Bharati, tell me what question do you have? Okay, some people are saying slow down, some people are saying fast. Okay, ma'am, clear. Thank you so much, and please remember Lepidopterans, Dipterans, and Coleopterans, and what are the group of organisms out there. That's all. Thank you, everyone. Okay. Now I will also explain the life cycle of Plasmodium, because I think that is also important and you might mess around there. So please, are we ready for? Okay. Are we ready for the life cycle of Plasmodium? Should I explain it to you? What say? Okay, I'll explain it to you. So listen, it goes like this. It's pretty much simple. Okay, so there is a Plasmodium, and Plasmodium, which is there, it is there inside the mosquito. You know what? Who causes malaria? It's not the mosquito, it's the Plasmodium. Mosquito is being framed, it's the poor guy. It's not so poor, but I am telling you, it's a medium poor guy. Okay, so basically what is happening over here is that the Plasmodium is basically growing up inside the mosquito, and it reaches up a stage which is known as sporozoite stage. Sporozoite stage. And this stage is found in the salivary gland of the mosquito. Salivary gland of mosquito, the M, right? Now the mosquito, when it comes and bites us, do you think it gives us blood? No, Baba, it does not give us blood. Basically, it takes the blood from us. And to take the blood from us very easily, it needs to put some anti-clotting things out there. And how does it passes? Through the saliva itself. So basically, through its saliva, it is passing that. And while it passes saliva, it also passes the sporozoite stage into our body. Now, when the sporozoite stage is now inside our body, it passes through our bloodstream and goes to the first camp. Remember, this is a terrorist and it has two camps. First camp is your liver cells. In the liver cells, my dear children, what do you call the liver cells as? You call them as hepatocytes. In the liver cells, these guys are going to get inside and are going to do reproduction, asexual reproduction. Remember, in human beings, there is no sexual reproduction happening of the Plasmodium. There is only asexual reproduction. Now, once they are asexually reproducing out there, basically, for once, they are going to burst open. Hepatocytes are going to burst open, and all these guys, the Plasmodium guys, are going to come out and mix with our blood cells. And now they have reached the second camp, which is what? Your RBC. This is the second camp. In the second camp, again, what are they going to do? Basically, in the RBC, they are going to divide themselves, and they are going to have a different stage of them. This different stage of them is known as gametocyte stage. What stage is this? Gametocyte stage. In this stage, it means that further, if at all they divide, they will be forming gametes. And these gametes can fuse together to form the other organisms. But do they form gametes in our body? No. I told you, no gamete formation, no sexual reproduction inside our body. So gametocytes are being formed over here. And not just that, they are inside our RBC. And why are they inside our RBC? They are feeding on our hemoglobin. And you know what they do? They break down the hemoglobin into heme and globin. And heme is very, very toxic for them. They are like, oh my God, it's very toxic. I have to convert into something else. So basically, they convert it into hemozoin for their own purpose. Therefore, their own purpose, they convert into hemozoin. So now we have hemozoin also over here. We have gametocyte of the Plasmodium also over here. Once the gametocytes, basically, if they are too much in number, this particular cell is going to burst open. And when they burst open, my dear children, all the gametocytes are going to be released, and also the hemozoin. And you know what? Hemozoin is toxic for us. When it comes inside our blood, my dear children, we get fevers and we get chills. And that is why we have occasional fevers and chills in this particular case. So hemozoin is the reason. Now, gametocytes are mixed over here. Gametocytes are basically mixed in our blood. Next time, the mosquito which will come and drink the blood will also carry these gametocytes. Now, these gametocytes are going to get inside the mosquito's body. I do not know how to make the mosquitoes. I'll make like this. Okay, so it gets inside the mosquito's body. Inside the gut of the mosquito, basically these gametocytes are going to form gametes, will undergo sexual reproduction and form ookinets and things like that later. So sexual reproduction is happening in the mosquito's body. And later one stage is being formed, that is sporozoite stage, where it will be moving to the salivary glands. And the same process repeats again and again. Okay, it repeats again and again. So that is how it is going to happen. So always remember, in which stage does it affect us? Sporozoite. What happens? What stage is being produced at the end? Gametocyte. Where does sexual reproduction happens? In the case of mosquito. Mosquito is also a host. We are also a host. Mosquito is also a vector for this particular disease. You have three types of Plasmodium: Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae, which causes malaria, but the malignant one is Plasmodium falciparum. Is it clear? Ayyo. Ma'am, low voice, increase please. Is everyone facing that? Is everyone facing the low voice? Or is it clear? Guys, is it clear, or everyone has some trouble in this? Shashank Gowda, is there no any antibiotic in our body? No, dear. It's basically, it's in the cyst, okay? It's inside a cyst. It is tough, resistant material. You might have studied in your class 10th grade. They do multiple fission. Okay, so everyone, if it is clear, can you give me a thumbs up? It will be really, really good. It's Jyoti. Yes, okay, ma'am. Thank you, dear. Your hair attractive, ma'am. I can't see even in the, sorry. Is it okay now? Okay. So, guys, if at all you have not clicked on the like button of this video, go click and subscribe to the channel. Also, and so do so many questions out there. Okay, so shall I go to the next chapter? Is it okay? If this is clear? Yes, now, okay, fine. So if it is clear, can I go to the next chapter that I have? So I was thinking, instead of doing sexual reproduction in flowering plants for now, I will be starting off with evolution, because this is a chapter that we have not touched the previous time. What do you say? Do you want evolution? Ma'am, you're so pretty. Thank you so much. You are also pretty. Okay. What do you say, guys? What should we do? It's upon you. Whatever you say, I am going to do it. Just a second, okay? Flowera. Sexual reproduction in flowering plants is what you want? Not evolution? Sure? Okay. So over here I have, thanks, evolution, flower not needed. Okay. So I think evolution is something that would be more, as in sexual reproduction in flowering plants I have covered previously. So if at all you watch the video, it will be really good. But okay, I can do the HOTS question. What should I do right now? So dear children, I think my charge is very, very less in the phone, and apparently I do not have charger over here. So just give me a quick second. If at all I can find someone to give me a charger, it will be good. Okay, just give me a second. Oh gosh. Bro, you're here? Can you give me the charger? Yeah, thank you. Okay, evolution. So we will do evolution. So I would have to change the settings a bit because my phone is okay. Listen. So I'll start with evolution. There are few concepts which have to be done. Then we can go ahead with the questions of sexual reproduction in flowering plants as well. Okay? Thanks, bro. Okay, guys. Okay. So let's get going. Evolution, the first question that we have, the first concept that we have is about the generations, as in basically the Homo sapiens, how they were formed, evolution of man. And this is one of the very, very important question. Okay, very, very important question that usually comes up. So first thing that you need to know is there were two types of organisms. One is Dryopithecus and one is Ramapithecus. Okay, Dryopithecus were basically ape-like. Ramapithecus, Rama, the term name is coming, no, from. So basically they were more man-like. So Dryopithecus and Ramapithecus were there. Dryopithecus were more ape-like and Ramapithecus were man-like. This happened 15 million years ago. Now, 3 to 4 million years ago, you have got man-like bones. Okay, man-like bones were found in Ethiopia as well as Tanzania. And it was found that the height was almost 4 feet. And they were doing bipedal locomotion, as in with 2 legs they were moving. Until now it was like this, but now they started walking like that. Okay, that is not too much important. We go to 2 million years ago. 2 million years ago in East African grasslands, there were Australopithecines. Okay, Australopithecines. This is important. They started using stone weapons, not for hunting animals. They were only eating fruits. This happened 2 million years ago. So for 15 million years ago, you got 2 million years ago, and that is where you got Australopithecines. And over here, they used stone weapons for hunting, and their main diet were fruits, not meat, nothing else. Okay, now just after that, you got Homo habilis, the first human-like organisms out there, the first human-like hominids out there you got. And when you talk about them, you mentioned their cranial capacity also. From this point onwards, you talk about their brain capacity. So it was 650 to 800 cc. Their cranial capacity was 650 to 800 cc. So I'll jot it down, the things that you have to remember, only that. Okay. 15 million years ago, 2. Dryopithecus, Ramapithecus. Ramapithecus were more man-like. Coming down from 15 to 2. 2 million years ago, Australopithecus. They were basically living in East Africa, and they started using stone weapons for hunting fruits, not for any meat. Now in the same time, almost around the same time, slowly, gradually, you got Homo habilis, Habibi, the first guys that came in. Okay, Homo habilis, the first human-like hominid, and basically they also were eating fruits and veggies, no meat. And their cranial capacity is also mentioned. It was 650 to 800 cc cranial capacity. Now, from Homo habilis came Homo erectus. And by Homo erectus, I mean they started standing erect. And it happened 1.5 million years ago. So from 2, let's directly jump to 1.5. And from this point onwards, my dear children, obviously their cranial capacity increased. From 800 cc, it has now become 900 cc. But they started eating meat. Okay, they started eating meat and they spread out everywhere. They went to Java, Indonesia, lot of places. Okay, Java is a place in Indonesia. They went to lot of places, they spread around. That is Homo erectus. Now from Homo erectus, you got two things. One is Neanderthal man, okay, and one is Homo sapiens. So Neanderthal man came first, and that was almost 1 lakh to 40,000 years ago. But they were the first and the special organisms with the highest cranial capacity, that is 1400 cc. And not just that, they started covering up their body with animal hides. They started burying their dead. They had some feelings with them. So always remember their features, because this is super important. They started burying their dead and basically covering their body as well. Parallelly, almost 75,000 to 10,000 years ago, you got Homo sapiens, that is the modern day human beings. And that's all. You do not need to remember anything else apart from this. Got it? Okay, did you get it? If at all you remember this, this is definitely going to help you out. Okay, so please remember this. So if you want, I can just show it to you like this. You have Dryopithecus and Ramapithecus. This happened 15 million years ago. They were ape-like, they were more man-like. Now coming down to what? 2 million years ago, you have Australopithecines, my dear children. They were in East African grasslands. They used stone weapons for basically having fruits and all. Now, just in this particular case only, just after that, they didn't mention the year, but they have mentioned about Homo habilis, the first human-like Homo habilis, the first human-like hominids out there. They were so special, their cranial capacity is mentioned, that is 650 to 800 cc, and it is clearly mentioned they ate only fruits, nothing else. Now, 1.5 million years ago, you got the next group of organism, that is Homo erectus. And Homo erectus, they started eating meat, and not just meat, the cranial capacity had increased to what? 900 cc, and also they spread to different places such as Java and all. Now from them came two of the things, that is, one would be what do you call Neanderthal man. Now this is super important because this have come so many times. They came around 1 lakh to 40,000 years back. 40,000 years back, their cranial capacity was 1,400 cc, the highest among all of them. They started covering their body. They started covering their body and also burying their dead. They started covering their body and burying their dead. Not just that, one more very important thing happened. Homo sapiens came almost 75,000 years ago to 10,000 years ago. They came, and my dear children, they were really good. Got it? Okay, got it? Thank you so much for giving the hearts. I hope this is clear. You will never forget it. Yeah? NEET aspirant, hello. Stanley Miller's experiment, yes, we'll talk about that also. So this is the first thing. Now next is comment on the similarity between thorn of bougainvillea and tendril of cucurbita. Remember, my dear children, thorn of bougainvillea, okay, and tendril of cucurbita, both of them are going to come from one particular phase, and that is known as axillary bud. It is known as axillary bud. So basically their origin is same. Origin is same, but function that they do is different. One is protection, one is basically what do you call for climbing purpose, for support purpose, right? So function is different, and whenever function is different, just understand they are homologous organs. What organs? Homologous organs, and homologous organ shows divergent evolution. So you might have seen HD. HD is basically homologous and divergent. Homologous and divergent, always remember HD. Okay, so divergent means from one common ancestor they had, but they moved to different location, and because they are in different location, they adapted well there, have different features out there. That is what you mean by divergent evolution. Did you get it? Hello dear. Okay, got it? Now there is one more very important thing. So this is the example for that, homologous and divergent. And one more important thing, CA or AC. So HD and AC, you can remember like that. Analogous organs and convergent evolution. What are analogous organs? These guys have not come from same origin, but they have same function. They look the same. For example, wings of butterfly, wings of birds. Wings of butterfly, I can do it like this, like it. I can break it like this, but don't do that, okay? But wings of birds, my dear children, we cannot do like that. But both of them are used for flying only. Do they have same origin? No, Baba. They do not have any origin. So what do you mean by that? Since they're staying in the same habitat, that is air, basically they have converged. They show convergent evolution. And basically the organs that you get after this is known as analogous organs. What do you call? Analogous organs. Okay? Ma'am, I want to get 70 on 70. You will get. Great job. Very hardworking. Thank you, Vani. Okay? Got it? So this is one very important thing that you need to remember. Now, sweet potato and potato, what is the relation? Sweet potato, potato, both of them are storing. But if at all you see sweet potato, my dear children, it is root modification. So over here, basically, suppose this is land. Okay, here you have root. Some roots basically store certain things. Some roots do not do anything, they are just there. Whereas you have stem. So some stem is just like this. Some stem basically grow down to store things. This is basically a stem modification. This is your potato. Whereas this is a root modification to store things, and this is your sweet potato. So both of them, do they have same origin? No. But they're staying in the same habitat, which is why they have to store food. So basically, same function is there. That means it is analogous organ, and they are showing convergent evolution. They are showing convergent evolution. Vaishnavi, Baba, what do you do not understand? Tell me. Yes, potato is stem modification. Ma'am, how to prepare for tomorrow's preparatory? Watch this live and also please solve questions. Okay, all of you. Vaishnavi, tell me da. What is the problem? Vaishnav, not Vaishnavi, I'm so sorry. I think that is what you do not understand. I do not understand. Tell what you're not understanding. I'm here to help you out. Okay, I think that's a spammer. With that I get that's a spammer. Hello, my friends, don't chat. Yeah, exactly. Okay. So guys, okay. Shall I go ahead? Okay, I shall go ahead with the next thing. Ma'am, your teaching is awesome. Thank you, Mardesh. Thank you so much, dear. Okay. Now we have named the evolution of any two plant part through geological periods from the tracheophyte ancestors. So over here, you need to remember this. So this is chlorophyte ancestors, tracheophyte, Rainier type, xylophyton. Now this you will only be able to remember. Sir, okay. Okay. So basically, you need to know one very important thing, and that is, listen to me. So basically, we had algae. In plants, we had algae. Okay, algae basically came under plants because they were able to do photosynthesis. That's all. They do not have root-like, stem-like, any feature. They do not have anything, leaf, root, nothing. From algae, you got bryophytes. Bryophytes were the one which had root-like, stem-like, and everything like features. They did not have proper things, but they had something like that. From bryophytes, they got pteridophytes. Now, pteridophytes had root, stem, leaves, everything. Apart from that, they also had one very important thing, and that is vascular system. Vascular system is there for transport of water as well as food. For water, they had the xylem. For transport of food, they have what do you call phloem. Okay, now listen to me carefully. Trachea is a part of xylem itself. So basically, it's a part of vascular system. So when I say tracheophytes, it's basically pteridophytes only. Pteridophytes only. Okay, a group of pteridophytes. Now, from pteridophytes, my dear children, they got pretty more advanced. You got gymnosperms. As in, they started producing seeds now. From gymnosperms, my dear children, you got angiosperms. Now, these seeds were covered with flowers out there. Okay, this is how things went. Algae, bryophytes, pteridophytes, gymnosperms, angiosperms. Take a screenshot of this, otherwise it might go. Okay, otherwise it might go. Please take a screenshot of this. This is important. Cool. Now, you have understood this flow. Now, when I bring it over here, you are asked, name the evolution of any two plants through the geological period from tracheophyte ancestors. So now the picture goes like this. Chlorophyte is algae. From algae, you got bryophytes, right? The bryophytes, let's see if they are there. Yes, these are the bryophytes. Where are they in this particular era? You have three era: Paleozoic, Mesozoic, Cenozoic. In the Cenozoic era, you have got the bryophytes. Now coming back over here, you got tracheophytes, that is pteridophytes. From tracheophytes, my dear children, some of them, they're developing. They're still in pteridophyte stage only. They are developing one by one. But in between, you saw Zoosterophyllum, arborescent lycopods, and herbaceous lycopods. This question has not been asked yet, but be sure. So from here, in Paleozoic era, you got arborescent lycopods, and herbaceous lycopods you got in Mesozoic era. Okay, if we forget this, totally fine. Okay, if at all we are not able to remember this, totally fine. Just remember one more thing. From the Rainier, they got Rainier type plants. From the Linear type plants, you got Xylophyton. Now from xylophyton, things are going to change. You get the modern day pteridophytes, and this is important. So modern day pteridophytes are what? Horsetail and ferns. Horsetail, that is Spinopsid, and ferns. Where are they? In the Mesozoic era. Where in the Mesozoic era? Jurassic and Cretaceous. Jurassic and Cretaceous is where these guys are there. They are modern day pteridophytes. And not just them. From the pteridophytes, from the older pteridophytes, you got gymnosperms also. Ginkgos, conifers. Knitales, cycads. CC over here, GG over here. Both of them coming in the Mesozoic era, especially in the Jurassic and Cretaceous period. This is super important question. They are gymnosperm. These four things are gymnosperms. Okay, these two are modern day pteridophytes. Everything happened in this particular time. Now if we go down a bit, from the progymnosperm, first a xylophyton, then the progymnosperms, then seed ferns, then here if at all you see, you got angiosperms. The first were dicots, then came monocots. Angiosperms completely came in Cenozoic era. Before that, in Cretaceous period, where GG and everything was there, dicots also came into the picture. Dicots also came into the picture. This question is important. This question is tricky. You might forget it. So you can tell name of two plants. One is angiosperms. So for angiosperms, trichophyte, then Rhinier type plants. Then you have xylophyton. From xylophyton, you have got gymnosperms, and then you have got progymnosperms, and then seed ferns and angiosperms. Okay, please write it. It is going to help you. And one is ferns itself. You make the flow chart, and that's done. Okay, genetic code topic, that is done, dear. Now it is Stanley Miller's experiment. For those of you who are asking, Stanley Miller's experiment was a proof to Operon and Haldane. So Operon and Haldane basically said that chemical evolution is happening. From the inorganic things we are getting organic, from the inorganic things we are getting organic stuff. What are the inorganic things that were there in the early environment? It was basically CH4, that is ammonia, CH4, that is methane, NH3, that is ammonia, H2O, that is water in the form of water vapor, and H2. Okay, these were the gases, and with this we are going to get organic things, materials. Is it true? Yes. And basically Miller tried to prove it. He had a closed flask-like structure, a closed chamber. Over here he had kept boiling water. The water kept boiling. When the water kept boiling, it basically released water vapors. Now water vapor was there. Now in the early environment there was very high temperature. How did he maintain the high temperature? With the use of electrodes. So he had kept two electrodes, and these electrodes made sure the temperature is super high. When the temperature was super high, these gases reacted, okay, and produces organic materials. These organic materials came and flowed up over here. What was the first organic material that Miller found? Miller found the first organic material, which was amino acids. If at all the question comes, did nucleic acid, anything was found by Miller? No, Baba. First thing in the Stanley Miller experiment that he got was amino acid, nothing else. Others did so many experiments and they got different things, but not this. Okay? Got it? Fine. Hello, neat aspirant. I am from Gujarat. Hello. Okay, so this is Stanley Miller's experiment, and remember, only amino acid you got. Now there is convergent and divergent evolution example of Australian marsupials as well as placental mammals. Do you need explanation for this? This is also an important question. So I'll go with Hardy-Weinberg first and then we'll come back to this, okay? So Hardy-Weinberg, I'll just explain it to you rather than explaining all this thing. So Hardy-Weinberg's law says the frequency of alleles is going to remain same throughout generation. Throughout generation to generation, it is going to remain the same itself. That means if I say that in here there are tall people more and the frequency of capital T allele is more, okay, and the frequency of small t allele is less, it is going to be the same generation, after every single generation. Do you think that is true? No, that is nonsense. Okay, so basically the frequency of alleles cannot remain same generation after generation. It is definitely going to change. And basically these factors that are going to affect the frequency of alleles is are going to be asked to you. And what are these factors? The first factor that you have is gene flow. Gene flow, or you also call this as gene migration, is a very important topic. It means that suppose tomorrow, okay, suppose tomorrow over here so many Americans came. Okay, so many Americans came. So the white people are going to be more in number, and so many Indians went there. So black people are also going to be there. The frequency of allele is going to be changed. The frequency of allele is going to be changed out there. You get it? When from one place to another place, people are going to move around. The frequency alleles that they carry, that is also going to move around, right? So that is known as gene flow or gene migration. Second one is genetic drift. Genetic drift. And by genetic drift, I mean basically suppose there is over here you have more green beetles. For some reason there are more green beetles and there are very less white beetles out there. So green beetles are saying, we are the one who are rulers over here, you guys are good for nothing. But one day one big elephant came and stamped over here. So what happened? All the green ones basically they died. What were remaining? The white ones were remaining. The allelic frequency, does it change? Yes. Has it changed randomly? Yes. This is known as, this random change which is going to happen is known as genetic drift. Okay, this is going to be known as genetic drift. Super important. Now next thing, you have mutation happening. Suppose over here you had green and white. Some mutation happened, you now have yellow also. Don't you think the frequency has changed? Yes. So mutation is one thing. Now, the next thing is genetic recombination. That means in sexual reproduction, we have studied the chromosomes which are there, they change certain things. Okay, they change the materials. You have newer combination. In that way also, you can get new combinations out there, right? Crossing over can also lead to a change in the allelic frequency. Then, you have natural selection. You have natural selection. One day, all the organisms who were having certain genes were selected. The other organisms were not. Why? Because of the nature. You have heard about industrial melanism. Okay, you have heard about industrial melanism. The white ones were dominating, the black ones not. Later on, the black ones were dominating, the white ones were not. Why? Because of the industries which were being formed out there, and the nature was selecting it. So this is an example of natural selection. Again, there is a change that is being happening. So these are the factors that you need to remember. When amphibian evolution, we'll come to that. Okay, we'll come to that also. So these are the things that you need to remember. Now in natural selection, you have three different types of natural selection which is usually being asked. Listen to me carefully. So suppose I have three scenarios. Suppose only tall and the dwarf, both of them are selected. Two extreme character is being selected. I'll call this as stabilizing selection. I'll call this as disruptive selection. Why? Because two extreme characters are being selected, tall and dwarf both. So it is what? Disruptive selection. Now over here, suppose neither tall nor dwarf, the middle character is being selected. The middle character. This is going to be stabilizing selection. Stabilizing selection. Now suppose over here, only one extreme character is being selected. Only the dwarf ones are selected. Not the tall, only the dwarf. Or either the tall one is selected, not the dwarf one. Extreme character is being selected. This is known as directional selection. Only in one direction it is being moving. This is known as directional selection. Okay, so there are three selection methods. You can be given the picture. You might be asked the question. Please go take the correct one. Okay, you might get numericals also. Please go through it. It's called destiny. Okay, got it? Now, one last thing that I want to teach over, second last thing that I want to teach over here is evolutionary significance of the following organism, that is shrews and dolphins. My dear children, always remember farm. What is farm? M for fishes, A for amphibians, R for reptiles, M for mammals, and over here you also have birds. Okay? So from reptiles you got birds as well as mammals. So fishes were the first one which were there in our vertebrates. Okay, they were there in the water bodies. Now slowly one fish was there which was having very thick fin. So it was able to move its fin and walk on the ground out there. And that's how you got basically the evolutionary significance of the lobe fins. So lobe fins were those particular fishes. They had, basically they're thought of like a link between fishes and amphibians, because you know amphibians are the one that live both in land as well as water. So they are the connecting link that we have between the fishes as well as amphibians. So if at all you are asked a question about lobe fins, please know this is very important to know. They mark a critical step in vertebrate evolution, bridging the gap between fishes and land-dwelling animals. These animals called lobe fins evolved into the first amphibians that live both on land. So they are considered to be the ancestors of frog and well as salamanders. Similarly, the reptiles basically gave down the mammals and the birds. Between the reptiles and mammals, the connecting link that you have is shrews. Is what? Shrews. So here you have lobe fins and there you have shrews. Remember. Okay. Samarth, hello dear, hello, hello, hello. A very big hello. Got it? Now, last question of this particular chapter, that is Australian marsupials and Australian placental mammals. Listen, over here, you might have heard about Darwin's radiation, adaptive radiation, right? So it was said that there were a group of organisms right over here, ancestors. He went to an island, he saw that group of ancestors over here. Some of them moved this side, some moved this side, some moved this side, some moved this side, some moved this side. They were what? Australian marsupials. Suppose they are Australian marsupials. Remember, all of them are there in the same geographical area. They moved to certain areas. They got better adapted there. Because of their changes, they got adapted there. But they had the same origin. Same origin, moving to a different area, getting adapted there. Divergent evolution. What is it? Diverging. They are diverging. Okay? But they have similar structure. It is an example of divergent evolution when you talk about Australian marsupials alone. But now suppose you are considering one more thing. Okay, you are also considering placental mammals. So placental mammals also came like this. Placental mammals also came into the picture. And the placental mammals, some of them moved this side, some moved this side, some moved this side, some moved this side, some moved this side. Basically, they also moved, right? If at all I am just talking about placental mammals, they are also showing divergent evolution. But if I talk about both of them together, these two organisms come from two different groups. But now they have converged to a point where they might have similar function. What do I say? When I keep both of them together, it is example of convergent evolution. This is going to be convergent evolution. Always remember this when both of them are equally considered. Got it? Now here the question that comes up is this. Australian marsupials and Australian placental mammals explain convergent evolution and adaptive radiation. How? So you have to say that in Australian marsupial, many have evolved from a common ancestor, moved to different location in a geographical area. This is adaptive radiation. Now similarly, placental mammals also came into Australia and moved into separate location, just like how were Australian marsupials moving, which is why they were converging into a point, because they were staying in the same habitat. And now you might be given this question that you have to match the following. Mole with what? So placental mammals, all the name is smaller, whereas Australian marsupials, their name are bigger. So mole, marsupial mole. Anteater, numbat. It's anteater only, different type. Okay. Mouse and marsupial mouse. Similarity you see. Lemur and spotted cuscus. So this is a question that came in the MQP also. Please remember. Flying squirrel, flying phalanger. Bobcat, Tasmanian tiger cat. Wolf, Tasmanian wolf. So all the placental mammal names are small, whereas this is going to be bigger. So the thing which is not contrary to each other is this, that is lemur and spotted cuscus. So please remember, lemur is from placental mammals and this guy is from this side, that is Australian marsupials. Similarly, from placental, anteater from this side and numbat from that side also. Remember this. Okay. So Darwin's finches is also an example of adaptive radiation. And now what you want? Explain in situ and ex situ conservation. Okay. So these are the other questions that you need to study from this chapter. Please make sure you study and then only go. Otherwise, you might be losing some marks out there. Okay. And in situ and ex situ, I can just explain. And my dear children, with that, probably Lifespan is the time period in which an organism will be living. Life cycle is basically the cycle of events that are going to happen from the birth to the death. Okay. So ex situ conservation means I am conserving an organism outside their natural habitat. For example, zoo. I have brought in some organism from other places and I put it in a zoo. This is ex situ. Similarly, botanical gardens. I have taken some plants from other places and put it over here. And over here, this year, you are asked mostly about the advanced ex situ conservation method. So advanced ex situ conservation are three of them. One is cryopreservation, where you are going to preserve the gametes out there in -190 degrees Celsius in the liquid nitrogen. The second one is IVF, in vitro fertilization, where the organism which you feel that might die, you are taking the gametes and basically fusing in the laboratory conditions so that they don't get extinct. The fourth one which is there is seed banks, where you are saving the seeds of the organisms which you think are in danger. Okay, so these are the advanced thing: seed bank, cryopreservation, IVF. Remember it. And also tissue culture. Four things are there. Tissue culture, organisms, plants which are going to die, you are tissue culturing them and increasing their number. In situ means you are conserving them in their natural habitat. For example, you have biosphere reserves, you have national parks, you have wildlife sanctuaries. Wildlife safari park is different. Wildlife safari parks come under ex situ. Wildlife sanctuary comes under in situ. Okay, love from Gujarat. Thank you so much. So my dear children, I think I would end up the live right over here because I understand you need to study. But I want to tell one very important thing, and that is this thing. Okay, listen to me very carefully. So I'm telling you again, for those of you who had missed, so today morning you will be getting a revision video. This will be of 1 hour duration. It will be very, very fast. Okay, just for a revision purpose, nothing else. So you can watch that video. Apart from that, if at all you feel that you have a lot to cover and it's too difficult for you, then I had already made videos and kept. Okay, one video that I did, if at all you are worried from the chapter sexual reproduction to human health and diseases. So I came live previously in the previous exam. Just a second. Okay, just give me a second. Okay, where is it? Yeah. Yes. So if at all you see this, okay, if at all you see this, second PUC Preparatory Exam Marathon Part 1. Over here, I had discussed the 1 marker to all the questions, 5 marker questions from the chapter Sexual Reproduction in Flowering Plants until Human Health and Diseases. So if at all you are not confident, please make sure you download the PDF. It will be there in the WhatsApp channel. Start solving the question. Watch the video if you have done any doubt. Start solving the question, watch the video. That PDF would be more than enough for you guys. Now, the rest of the chapter, microbes in human welfare until biodiversity and conservation, you should not be missing it up. So I did a recorded video for that. Okay, and the recorded video you can find in video section, not in live, in the video section. So this is going to be part 2. I'll show you that to you so that there is no confusion. So just give me a second. Where is it? Come on, come on, come on. Okay. I think it's taking time to load. Just give me a quick second, okay? It is definitely... Okay. Okay, so... Over here, if at all you move a bit down, we did for preparatory 1. Now the preparatory 2 is similar, is not basically going to change up. It's going to be the similar itself. So basically preparatory 2, yes, this one, as you can see, part 2 of the same thing is there. Refer to the questions and start solving it. It's really, really important for you to know that. If at all you do not know it, it will be a big trouble. So start solving it up. Okay? Fine. If it's too much time for you, PDF, refer PDF, and I said, solve the questions along with me if you have any doubts. Now there is a fixed question also. listen, the concepts of it is being repeated. It's not like the same question will be anyway coming. If there is any teacher who's fooling you and telling that this question will definitely come and there is no chance, there is no other question that will become, you are misguided. So do not fall under any of this trap. You have to basically solve the questions, understand the concepts, solve maximum number of questions which have usually come. So please do that. Okay, got it? So this is one very, very important thing. And yeah, that's all. Tomorrow morning, I have I have put up a video, would be coming up. It will be for revision. Please watch that video as well. It is going to definitely help you out. Okay? So I would be covering the most important chapters in that video. Works? Ma'am, I'm watching that recorded live video. I have completed that video and along with the other 3-hour video. Will I be able to complete it within 6 to 5 hours? If you think, what time is the revision video, ma'am? It will be coming in at 5 o'clock in the morning. If you want, we can put it at 4 o'clock in the morning also, if you want. Okay? I'll change the time to 4 so that it will be useful for you guys as well. So I know you have to do your own studies, which is why I'm ending up the live over here. Thank you so much, Slim Shade. Okay. So Sushila, as I said, dear, please, if you think that watching a video is difficult for you, please take up the WhatsApp channel. I'll just put the PDF once more so that it will be easy for you to revise and go through it. That will be enough. So 2 PDFs will be given, part 1 and part 2. Just go through it. Okay? I hope that makes sense. Fine. So thanks a lot, dear children, and I wish you guys succeed and you guys get good marks. I'm praying for that. Do not be worried if at all there's too much left. Sleep for some time, and you guys are absolutely awesome. Always remember that. Okay, there should not be any doubt in that. And yeah, that's all. So I'll end up this live session right there, and thank you so much, Sushila. Thank you for everything. Prajwal, thank you so much, dear. Everyone, thank you so much, and do not forget to subscribe to the channel and click on the like button as well. See you soon. Wishing you the best, and go study. Go study by your own. Okay? So, bye-bye. Take care. Lots of love. Tharva, thanks a lot, ma'am. Very good class I had today. You're most welcome. Good night. Good night to you, Ullas. Okay, Riaz. See you, children.